Long press. Living beings. And second, it should not be present in any non-living. If both these criteria are fulfilled, then only we will say that it is all-inclusive defining property of living beings. Now let us try to see each of these criteria on the basis of these 2 points. Okay, so let us first of all go to growth. Are both these points clear to everyone? Now coming to growth. See here. Mountains, boulders and sand mounds do grow. It means growth cannot be considered as an all-inclusive defining property of living being because certain non-living things are also showing growth. While we have said that the all-inclusive defining property is that no non-living should show this property. So growth as such is not a defining property of living beings because it is also shown by non-living things. Okay? But there is a difference. There is a difference. Growth can be extrinsic and intrinsic or internal. Extrinsic growth means growth due to deposition from outside. That is called as extrinsic growth. And intrinsic growth means it is growing from inside. So suppose there is a mountain. Suppose there is a mountain or a sand dune and more sand deposits on it from outside. It will grow. But this is extrinsic or external growth. But we, living beings, we show intrinsic growth, growth from inside. We do not paste any muscles from outside to make the six-pack ab. Okay, our growth is from inside. So our growth is intrinsic growth. So although both living and non-living show growth, but there's a difference. Growth of living beings is always intrinsic or internal growth, while growth of non-living things is always extrinsic or by external deposition. So now let us see. If we simply say growth, then growth is not a defining property of living beings. But if we say intrinsic growth or internal growth, then it is a defining property of living being. So condition under which growth is taking place, if this is also mentioned, then it becomes a defining property of living beings. Let us see here. In living organism, growth is from inside. Growth, therefore, cannot be taken as a defining property of living organisms. Conditions under which it can be observed in all living organisms have to be explained, and then we understand that growth is a characteristic of living being. Okay, so what is that condition? Condition is intrinsic or internal growth. Fine. So first thing is regarding growth. Next we come to reproduction. Now again you see, reproduction can be asexual reproduction where there is no gametic fusion, or it can be sexual reproduction. Now reproduction, there are two things that no doubt that no non-living shows reproduction. This criteria it is fulfilling, that no non-living shows reproduction. But do all living beings show reproduction? Or we can differentiate reproduction and growth? This is the problem. Suppose there is a unicellular organism. Suppose there is a bacterial cell. It divided, it underwent cell division to perform growth. But simultaneously, reproduction also took place. So in unicellular organisms, growth and reproduction are mutually inclusive properties. We cannot differentiate between growth and reproduction in unicellular organisms. That is why first reason why reproduction is not considered as a defining property of living being, because for unicellular organisms we cannot differentiate between growth and reproduction. They are mutually inclusive phenomena. Second thing is, children, if you see that there are many things which do not show reproduction at all. For example, you might have studied the life cycle of honeybees. In honeybees, cellular organization and metabolism are all inclusive defining property of living beings. Now the question arises, suppose I take a test tube and in this test tube I add some protein and then I add some protease enzyme. Will this protein be digested or not? Yes, this protein will be digested. Then you can say, sir, this test tube is also showing metabolism, so this test tube should also be considered as a living being. So here, children, isolated metabolic reactions performed in vitro are very much living reactions, but they are not living beings because the test tube is not showing a cellular organization. Also, a metabolic reaction can be performed in the test tube. So remember that isolated metabolic reactions performed in vitro are living reactions, but not living beings. Okay, fine. So let us see this point. First of all, cellular organization of body is a defining feature of life forms. Next. All plants, animals, fungi, microbes exhibit metabolism. Okay, and no non-living object exhibits metabolism, means metabolism is fulfilling both the criteria. Next is, hence, while metabolism is a defining feature of all living organisms without exception, isolated metabolic reactions in vitro are non-living things but surely living reactions. Okay. So this is all regarding the metabolism. Next, we are coming to the last, and that is consciousness. Consciousness. Okay. You see, all living beings, they can perceive their change in their surrounding environment and accordingly they can respond. Remember mango. You like mango fruit very much, but why don't you get the mango fruit throughout the year? Tree of the mango is always there, but fruit is formed only in that some plants, they require shorter photo period or light period to flower. Some plants require longer photo period to flower. So this is a response to change in environment. Okay. Suppose some a red hot stick is brought near your hand. Even the moment it will touch, although your eyes are not there, you will just immediately move your hand. Why? Because you are responding to that change. So remember, consciousness is an all-inclusive defining property of living being. From bacteria till human being, everyone shows consciousness. And no non-living shows consciousness. So again, consciousness is a defining property of living organism. Okay. Now one thing is that suppose a person is brain dead and he is in coma and he is lying in the hospital. What to call? You see, in such case, it is very difficult to define, neither living nor non-living. So if the person brain dead normally do not come back alive. Okay, but anyway, so this cannot be said living or non-living. This is a twilight zone where the person is there. Fine. Okay. Next thing is, what I am doing, whether it is correct or incorrect, I should do it or not show it. This is called as self-consciousness. So consciousness is present in all living beings, but self-consciousness is present only in human beings. Remember this point. This consciousness is present in all living beings, but self-consciousness is present only in human beings. That's all. Fine. So we conclude this part of the chapter by all these points. Fine. Next, we come to the second part of the chapter, that is diversity in the living world. Right? Second part of the chapter, diversity in the living world. So there are certain definitions which we have to see in diversity in the living world. We are starting with the first definition, that is biodiversity. Okay. So refers to the number and type of organisms present on Earth. Okay. So number and types of organism. So number means how many species are there, and types means whether which are prokaryote or eukaryote, unicellular or multicellular, autotrophic or heterotrophic, terrestrial or aquatic. These are the different types, and total number is a number. Fine. So till now we have been able to identify 17 to 18 lakh species of living beings, that is 1.7 to 1.8 million. But remember, this number is not fixed. More and more voyage is done by different scientists throughout the world, in the ocean, on the rocks, mountains, everywhere. And every year, many new species are explored. Okay, many new species are explored. So as the time will pass, this number will keep on increasing. Of course, some of them may become extinct also in the due course of time. But more and new species will be. Okay, not only that, sometimes this also happens that if you explore the already explored area, if you re-explore it, sometimes some of them which we have failed to recover, this time we are able to find them. So not only in new area new species can be found, if old area are searched again, sometime there also new species are found. Fine. So this is called as the biodiversity. Now, small water break. Someone is asking, sir, we can say 13% of total. How can you know what is the total number? Someone says total number is 7 lakh, someone says 13 lakh, something which is hidden. How can you say? What's your name? Bhanu Satya Sheikh. Because we do not know the exact number what is present, and how can anyone tell what the exact number? So we cannot say that 13 or 14%, because the speculation is very wide. From 7 lakh to 15 lakh species are present, people say. Fine. Okay. So children, now have got 17 to 18 lakh species of different types of plants, animals, microbes, etc. So one single person cannot study all this. So we need to have a specific grouping of organisms so that the study becomes easy. But for that grouping, we will have to first describe the organism. Okay. And identify which organism we are talking about, and then we will have to give it a name. Okay, we will have to give it a name. So first of all, we will describe an organism. Suppose you found a plant, you will say how is the leaf, how is the fruit, how is the flower, how is the stem, how is the root. Then you will cut a section and you will tell what is the anatomy of leaf, what is the anatomy of root. Then you will study the life cycle, whether it is producing fruits or not, whether it is producing seeds or not. All these features you will note down. This is called as the characterization, okay, or description. Then on the basis of this, you will try to identify which organism is this, okay. Then you will classify which group I should place it, and then you will have to give it a name, okay, because name, and then you will do classification. Fine, okay. So this is identification. So first of all, there will be description and there will be identification, see, and then you will give a name. This naming of organisms is called as nomenclature. Now remember, today man is scientist also, but there was a time man was not a scientist. But that time also, man was in close interaction with living beings, plants, animals, for food, fodder, shelter, fiber, medicines, and number of things. So naturally, when man was not a scientist and he is using living beings, he would have given them some name. And these names are called as vernacular names, and they are given in the local language. They are given in the local language. But when man became scientist, he realized that this is a problem, because language changes every 30, 40, 50 kilometers. So same organism is being known by 100 different names in different parts of the world. So it is very difficult to discuss about the same organism in different parts of the world. Okay. These names, which are called as the vernacular names, they are non-standardized, means there is no standard name. For example, same thing here, you call it Koperica. Okay, in Hindi it is called Nariyal, in English it is called coconut, some places it is called copra, and maybe hundreds names. So if you have to study, then it is a problem. Okay, so that is why man decided to give a standardized name, that one organism is known by the same name throughout the world. And then came the scientific naming of the organism. And for this naming, first of all, you will have to identify the organism, which organism I'm talking about. And for identifying the organism, first you will have to describe the organism. So describe the organism, identify the organism, name the organism, and then classify the organism where you want to put it. Fine. Okay. Now coming to naming. Now if you've decided that one organism will be known by same name throughout the world, then you will have to have a standard organization. Okay. For example, suppose cricket is played in many countries of the world, but the rules are same for every country. Any two countries or even within a country, cricket is being played, the rules are same throughout the world. Why? Because there is an organization called as International Cricket Council, ICC, which makes rules which every country has to follow. Similarly, codes were made for naming. So first code that came for naming was of the plants. International Code for Botanical Nomenclature. So it was decided that this code, this organization will decide what will be the standard name of plants which will be followed by all the scientists throughout the world. When ICBN was made, then zoologists said we will also follow, and the next one, International Code of Zoological Nomenclature, was made, which decides about the naming of the animals. So ICBN, International Code, this question is very frequently asked. International Code for Botanical Nomenclature for naming of the plants, and International Code of Zoological Nomenclature for naming of animals. Fine. Now, the question was, what rules should we follow? What rules should we follow? So many people gave their proposals. Some came with name of organism consisting of many words. Polynomial system it is called. Okay. For example, Calophyllum, Saxatilis, Folis, Graminis, Umbilitis, Corymbus. This was name of one plant. Okay. This was called as polynomial system. But this was not practical. How can you remember so many words for one organism? Then came a trinomial system, which is even today followed in some cases, where a scientific name consisted of 3 words. Okay. Name of the genus, name of the species, and name of the variety in case of plants, and name of subspecies in name of animals. So some places it is still being used, but the most common and the most accepted and the most popular, that was the binomial nomenclature. And this binomial nomenclature was given by Carolus Linnaeus. So you have to remember, the most followed nomenclature system is the binomial nomenclature, and this binomial nomenclature was given by Carolus Linnaeus. Okay, and this binomial nomenclature has 2 components. First component is the generic name or name of the genus, and second component is the specific name or the name of the species. Now this binomial nomenclature also has certain rules. So what are the rules of binomial nomenclature? Let us try to. This is, children, very, very important. This part from these questions are very frequently asked. Fine. So now these are the rules of binomial nomenclature. See, first of all, scientific names are always given in Latin. And if it is derived from some other language, it is Latinized, means converted to Latin. The reason is, children, because Latin is a dead language. So there is no chances of any change. Once a name is given, it will never change, because Latin is a dead language. Second advantage of Latin is that Latin has no synonyms. So one word has only one meaning. So in Latin, there is no chances of any confusion between 2 words. Okay, fine. Because one word has only one meaning, no synonym. So first thing, it is given in Latin. Second, the first name biological represented genus, and the second name is the species. And after the name of the species, the author who has given this name, his name is written in abbreviated form. So generic epithet, specific epithet, followed by the name of the author in abbreviated or short form. Fine. Third is, both words in a biological name when handwritten are separately underlined. This is a fault which many of us do. We write the name, we don't underline. It is wrong. So name of the genus and name of the species should be separately underlined. And when it is being typed or printed, it should be printed in italics. Okay, italics letters, I will show you. It should be printed in italics. Why we do this? We do this to show the Latin origin of word, that this word has originated from the Latin language. That is why we do this. Okay. Now the name of the genus starts with a capital letter and the name of the species start with small letter. Fine. But children, one thing you have to remember, that when you are underlining name of genus, you will underline, name of species you will underline, but name of the author you will not underline, because author's name cannot be converted into Latin language. So this thing we have to keep in mind. Now let us see the mango. If you see here, mango generic name, this you can see the difference in this printing and this printing. This is italics printing. I will slightly enlarge it. Fine. This is italics printing. See, generic name and specific name are printed in italics. Name of genus starting with capital, name of species starting with small. After this, you will write name of the author, Linnaeus. Okay. Then it is complete. See here, it is written Mangifera. Okay. See, Mangifera indica, and after that Linnaeus in short form Lin. So this is the correct way of writing the scientific name according to the binomial nomenclature. So this is completed. Now coming to classification. Now let us try to understand classification. Classification is grouping of organisms into convenient categories on the basis of their observable characters. Okay. Grouping of organisms into convenient categories on the basis of their observable characters. Now, what is the advantage of classification? See, you see in your home, in your almirah, if you have kept 30, 40, 50 books and you want to find one book, it takes you 15, 20 minutes, you will throw it like this, and you find, and after 15, 20 minutes, you may find the book you wanted. But you go to a library, there are thousands of books in a library, but the book you want, you get within 5 minutes. How? Have you ever thought? Because you have dumped your book randomly. While library has kept the book in a catalogued form. For example, first few almirahs, 100 almirahs will be only for the books on science. Next 100 almirahs will be books only on the humanities science. On science also, they have decided that the first 5, 10 almirah will be of biological science, next 10 will be of physics, next 10 of chemistry, like this. And in the first 5, 10 almirah of biological sciences, they have also decided that first few almirah will be exclusive zoology, second few almirahs exclusive botany, and then the common topics. This way, immediately you want to get a plant physiology, you know that plant physiology book will be in the first 2 almirah, and immediately you will find it. So this grouping makes the study convenient. That is why classification is done. So classification is the process by which anything is grouped into convenient categories based on some easily observable characters. Okay. For example, I am giving you one example. Suppose you have never seen a plant. You have never seen a plant, but I am just describing the features of plant and you will tell that classification of that plant. I am saying this plant has taproot system. It has reticulate venation of leaves. You will immediately, sir, it is and it is producing flowers. Okay, and morphology I have told you. You will say 2 things. First, sir, it belongs to division angiosperms and it belongs to the class dicotyledonary. Why? Flower producing division angiosperm and reticulate venation of leaves and taproot system, it means it is a dicot. So division and class you have told without seeing the plant. Then I say, in this plant, the ovary is obliquely placed, placenta is slightly solemn, there are 5 sepals, 5 petals, and 5 stamens, and stamens are epipetalous, and the gynoecium is bicarpellary syncarp superior ovary with axile placentation. Immediately you will shout, sir, you are talking about Solanaceae family. So you got the family of this plant while you have never seen this plant. So this is the advantage of classification, so that if you have classification, even without seeing any organism, just by its description, you come to know that which organism we are talking about. So it makes the study very, very easy. So this is all regarding the classification. Now comes a term taxa. This you need to explain because this is important. What is a taxa? And taxa, plural of taxa is taxon. So there is not much difference between taxa and taxon. Okay, taxa. Okay. For example, if I say genus, okay, genus, or if I say species, suppose species, you will not get which organism we are talking about. This is a category. But if I say potato, tomato, makoi, these are all real biological objects. You know what we are talking about at the species level. They are different species of the same genus. Okay, so they are taxa at the same category. Suppose we say class. Okay, class. Class is a category. But if we say mammals, we say dicots, then these are real biological objects at the class level. So mammals and dicots are taxa at the same level. Okay, mammals and dicots are the taxa at the same level. Here you can add dog, cat. Okay, so these potato, tomato, makoi, dog, cat are taxa at same level. Mammals, dicots are taxa at same level. But dog and mammals are taxa at different level, because dog is the taxa at the genus or species level and mammals are the taxa at the class level. So this is the difference between the category and the taxa. Let us try to see this. See. What image do you see when you think of a dog? Obviously, each one of us sees dog and not cats. Now, if you think of Alsatian, Alsatian is, children, a breed of dog. Okay, a very powerful breed of dog is called Alsatian. We know that we are talking about, similarly, suppose we were to say mammals, you would of course think of animals with external ears and body hairs. Likewise, in plants, if we try to talk wheat, the picture of each of us mind will be of wheat plant, not of rice or any other plant. Hence, all these dogs, cats, mammals, wheat, rice, plant, animals, etc. are convenient categories we use to study an organism. The scientific term for these is taxa. Okay. Hence, you must recognize that taxa can indicate categories at very different levels. This point is important, that taxa can indicate categories at different levels. Plants also form a taxa. You see, the highest level of taxa, category will be kingdom and the taxa will be plants. Category will be kingdom, taxa will be animals. Okay. Fine. Someone is saying that explain this line that properties of tissue are not present. You tell me whether individual cell of your any organ will perform the same function as the organ will perform. You tell me that whether single cell of xylem can perform the same function what xylem performs. Okay, so that is why the tissue will perform as such, because tissue are many types of cells. So individual cell will not perform that function which the tissue will perform. That's all. Fine. Okay. Anyway. So here we must animals, mammals, dogs are taxa, but at known that dog is a mammal and mammals animal. Therefore, animals, mammals and dog represent taxa at different level. Very important. Animals are taxa at kingdom level, mammals are taxa at class level, and dogs are taxa at the genus level. Fine? Okay. So children, with this we complete this. Last thing, taxonomy. Now, study of taxa is taxonomy. Study of taxa is taxonomy. So taxonomy, there are 4 parts. First of all, description, then identification, then classification, and then nomenclature. Let us see. It is not a specific sequence, but these 4 are the parts of taxonomy. Okay. See, description or characterization. On the basis of this, we will identify, and on the basis of this, we will classify. But for that, you will have to give a name. So these are the parts of taxonomy. And to study taxonomy, we should know morphology, anatomy, reproductive details, evolutionary, everything we should know. Then we can only study the taxonomy. Last thing we are coming is systematics. Systematics. Systemic arrangement of organisms is called systematics. The term was given by Carolus Linnaeus. Okay, systematics term was given by Carolus Linnaeus in his book Systema Naturae. Fine, in his book Systema Naturae. And again, it is a Latin word. Systema Naturae means systemic arrangement of organisms. Fine. Now, systematic basically studies with relationship among organism and diversity. How far 2 organisms are close to each other and from where they separate from each other. So this relationship and diversity among organisms, this is called as the systematics. Later on, the scope of systematics was increased to study the phylogeny, that is the evolutionary significance. So today systematics very much depends upon the evolutionary studies or evolutionary relationships. So let us see these points. The scope of systematics was later enlarged to include identification, nomenclature, and classification. Systematics takes into account evolutionary relationship between organisms. Okay, fine. So with this, we complete this portion. Last, we come to taxonomic categories. Taxonomic categories are just groupings. Okay, a lower grouping, then higher grouping, then higher grouping, then higher grouping. For example, basic unit of classification is okay. Someone is suggesting we go faster with this topic. Okay, thank you, sir. Thank you so much. Okay. Okay. So, taxonomic categories means there is a hierarchy of groupings. Okay. Lowest grouping is species. Above that, there is genus. Above that, there is class. So, a group of related species is called genus. A group of related genus is called as the family. A group of related family is called as class. A group of related class, order. A group of related order is called as class. A group of related order is class is called as division in case of plants, and phylum in case of animals. And the highest most is the kingdom. Okay, fine. Okay. Last thing is to learn that as we go up in the taxonomical hierarchy from species up till kingdom, the number of common characters among members keep on decreasing. So classification, grouping of organisms in the same category at higher level becomes much and much difficult. Okay, fine. So let us see this thing. Say this point, very, very important. Lower the taxa, more are the characteristics that the members of within the taxa share. Higher the category, greater is the difficulty of determining the relationship to other taxa at the same level. Hence, problem of classification becomes more complex. So this question, this thing you have to keep in mind. Okay. And then please remember this chart. This chart is very, very important. Okay. Please remember this chart. This is very, very important. You should completely remember this chart. Question may be asked from this chart. Taxonomical aids are no more a part of your syllabus. So with this, we have completed our first chapter, that is the living world. And next we come to biological classification. Okay, biological classification, children. From the very early time, man had made attempts to classify organisms into different groups. So, but these earlier classification systems, very good question someone asked, that what do we mean by categories are distinct biological entities, not merely morphological aggregates? Sir, once briefly. Very good. So this is a question, children, that categories are distinct biological entities, not merely morpho aggregations, or once briefly. Yes. Remember that if we make grouping only on the basis of morphology, then all trees will come in the same group. It means pinus and mango will come together because both are trees. And herbs and shrubs will be different from trees. So, garden pea, okay, garden pea and mango will be far away because garden pea is a herb and mango is a tree. Is it correct? But it is not the case. It means when we have grouped the organism, we have not grouped only on the basis of morphology. We have studied all the aspects of that, and then we saw that although mango is a tree and pea is a herb, but mango and pea are much closer because both produce flowers, so both are angiosperms. Both have two cotyledons in their embryo, means both belong to the same class, dicotyledonary. While gymnosperms, while Pinus is not producing flowers, so it is not an angiosperm, but it is producing seed. It means it is a gymnosperm. So that is why this grouping is not on the basis of morphology only. This grouping is on the basis of all aspects, that is morphology, anatomy, embryology, and everything, and then grouping has been done. I think you have understood. Radhika Reddy. Okay, fine. That comes for the second water break. Hi. So, when living beings were grouped, it was said, so I said when man was not a scientist, then also he grouped organisms. But those earlier grouping were need-based, requirement-based, for fodder, which will give fodder, which will give shelter, which will give food, like this. But as our knowledge increased, people started grouping organisms on the basis of their observable characters, that is scientific. And we said that first scientific classification of plants was done by Aristotle. Was the earliest to attempt a more scientific basis of classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. And he also divided animals into two groups, those which have red blood cells and those that did not have red blood cells. So these were the first, most primitive type of scientific classification. But a more advanced type of scientific classification was given by Carolus Linnaeus, and he divided all living beings into two kingdoms. First was kingdom Plantae and second was kingdom Animalia. And there was only one criteria. There was only one criteria, cell wall. If cell wall is present, it has to be plant. What is the chemical nature? Nothing, but cell wall present. And if cell wall absent, it has to be animals. So there were only two kingdoms, that is cell wall present plants and cell wall absent animals. And this proceeded for a long period of time. But as our knowledge increased, we came to know the shortcomings of this system. So let us see what were the shortcomings of this two kingdom classification. See, first, earlier classification system included bacteria, blue-green algae, fungi, mosses, fern, gymnosperm, angiosperms under kingdom Plantae. The character that unified this whole kingdom was the organism included had a cell wall in this cell. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae with other groups which were eukaryotic. So first, there was no differentiation of prokaryotic organism and eukaryotic organism. Second, it also grouped together unicellular organisms and the multicellular ones. Okay, there was no differentiation between unicellular and multicellular organism. Chlamydomonas is unicellular, Spirogyra is multicellular. Both were placed under algae in the kingdom Plantae. So this was so first, no differentiation of prokaryote and eukaryote. Second, no differentiation of unicellular and multicellular organism. Okay. This classification did not differentiate between heterotrophic and autotrophic. Third, no criteria was taken as mode of nutrition, which today is a very, very important criteria, mode of nutrition. Okay, heterotrophic, autotrophic. Heterotrophic also saprophytic, parasitic, symbiotic. Autotrophic, chemoautotrophic, photoautotrophic. This is a very, very common, important criteria which was not discussed at that period of time. Fine? Okay. So next is chemical nature of cell wall. Even if you place that all organisms having cell wall will be in the same group, then also there is a problem, because the chemical nature of cell wall of bacteria is different. Chemical nature of cell wall of fungi is different, it is chitin, and the chemical nature of cell wall of the plants is different, it is cellulose. Fine. So these were the basic shortcomings of this two kingdom classification. Then in between came three kingdom and four kingdom also. I will not discuss much detail of that. Three kingdom tried to remove the problem of unicellular and multicellular. Okay. There is a problem with Chlamydomonas, that it is mentioned in both Plantae and it is mentioned in both Protista. So children, a basic thing I am saying, okay, if you are given a question, Chlamydomonas, as per the five kingdom classification, Chlamydomonas and Chlorella belong to which kingdom? And then you have option of both Protista and Plantae, please tick Protista. But if there is a question, which of the following is an alga? Or which of the following is a green alga? And you have Polysiphonia, Gracilaria, Fucus, and Chlamydomonas. Then what will you do? Polysiphonia, Gracilaria, Fucus, and Chlamydomonas, and they are asking which of the following is a green alga. Then certainly you will have to go for Chlamydomonas. But if you are given option between Plantae and Protista, then go for Protista. I think that is all you can understand from here. Fine. Okay. So in between came three kingdom also. Three kingdom classification separated all the unicellular organisms and placed in a separate kingdom, Protista. But this Protista of three kingdom was faulty because it contained both prokaryotes and eukaryotes. Then came four kingdom classification, which tried to remove this problem. So in this four kingdom classification, a separate kingdom Monera was made for the prokaryotes. So all prokaryotes kingdom Monera, all unicellular eukaryotes kingdom Protista, and then two Plantae and Animalia. But still there was a problem, because up till four kingdom classification, the fungi were present in kingdom Plantae. But if you see, there is no similarity between fungi and plants. Fungi have chitin cell wall, plants have cellulosic cell wall. Fungi are photoautotrophic, plants are photoautotrophic, while fungi are completely heterotrophic. Plant, their reserve food is mainly starch. In fungi, the reserve food is oil and glycogen. So that is why it was thought that fungi can no longer remain in the kingdom Plantae. So then came the five kingdom classification. okay, which separated fungi from plants. Okay, and thus how this chart is there for 5 kingdom classification. Kingdom Monera, all prokaryotes. Kingdom Protista, unicellular eukaryotes. Kingdom Fungi, multicellular eukaryotes, but they are heterotrophic and they do not have a tissue level organization. Plants, multicellular photosynthetic autotrophs, okay, animals, multicellular photosynthetic autotrophs having tissue level organization and phagotrophic mode of nutrition, although they are also heterotrophs. But fungi have absorptive mode of nutrition. They first digest the food in the external environment and then absorb. So these are the 5 kingdoms of classification. Fine. All these points are very, very important. This entire table is very important. And the 5 criteria for kingdom classification are: phylogeny means evolutionary relationship. So these are the 5 criteria of the 5 kingdom classification. Fine. Okay. Now, according to 5 kingdom classification, I said there are kingdoms: Monera, Protista, fungi, Plantae, and Animalia. Fine. If you see one thing, that starting from 2 kingdom to 5 kingdom, the 2 kingdoms, Plantae and Animalia, exist in all the kingdom system. Okay. 2 kingdom also, 3 kingdom also. So Plantae and Animalia, these 2 kingdoms are fixed from 2 kingdom up till 5 kingdom. The difference in other groups only. And as our knowledge will increase, again there will be changes. This is not fixed, and changes have already come. Okay. Carl Woese has proposed a 6 kingdom classification because he saw that in kingdom Monera, all members of kingdom Monera are called as bacteria. He saw that the chemical nature of cell wall of all bacteria is not similar. There is a group of bacteria which have non-peptidoglycan cell wall. And these came to be known as archaebacteria. And peptidoglycan cell wall, this known to eubacteria. Fine. So he said that these two cannot live in the same kingdom. So he gave a 6 kingdom classification where he broke the kingdom Monera into 2 kingdoms, archaebacteria and eubacteria. So now they became 6 kingdoms. Okay. He gave a higher level also. He said all eukaryotes in the domain eukarya. archaea, and eubacteria in the domain bacteria. So these 6 kingdoms were classified into 3 domains. Fine. So this is the 6 kingdom classification given by Carl Woese. A small mention of this is in your NCERT, that is why I told you. But our further study will be based on the 5 kingdom classification as proposed by R.H. Whittaker. So let us see. We are starting with kingdom Monera. So all the members of kingdom Monera are collectively called as bacteria, and these bacteria of different shape, size, you can see here, coccus, bacillus, vibrio, all that, fine. Now, next is, this line is very important. Though bacteria structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. 2 metabolic features I will tell you which are shown only by bacteria, by no other group of organisms. One of them is nitrogen fixation. No eukaryote has the ability of biological nitrogen fixation. And second, chemoautotrophic mode of nutrition, where an autotroph does not use sunlight, it oxidizes some inorganic compound and the energy released by this is used for food synthesis. So these 2 are exclusive metabolic features of only monerans, only bacteria. That is why NCERT has written, as a group they show most extensive metabolic diversity. They can do what others can do. They can do that also what others can do. But they can do certain things which others cannot do. So that is where their metabolic diversity is very, very high. Fine. Okay. Now, coming to mode of nutrition, see that most of the bacteria are heterotrophs, but some are autotrophs also. Now, again I said this kingdom Monera can be divided into 2, archaebacteria and eubacteria. Archaebacteria differ from other bacteria mainly in the chemical nature of the cell wall. Okay? Basically, the chemical nature of the wall, having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. So they can live in such extreme conditions like very hot temperature, okay, very acidic temperature, okay. So they can live in such conditions, anaerobic, completely anaerobic condition. On this basis, these archaebacteria are of 3 types: methanogens, which oxidize organic compounds, which break organic compounds and produce the methane gas, okay, and this methane gas is used as biogas. And in the rumen of cattle, for when cellulose metabolism is done, it is done by methanogens. That is why in human welfare chapter, when you study the biogas production, we use the gobar, that is the cow dung, because cow dung naturally contains the methanogens. Fine. Okay. Next is halophils. Halophils means halo word where comes, it means salt. So they'll grow in extremely salty conditions where other organisms cannot grow. And third is thermoacidophils. Thermoacidophils means temperature as high as 80 degrees centigrade and pH as low as 2. So highly acidic condition and high temperature. This is found in hot water springs. Okay. If you go to mountains, there are certain places where very hot boiling water comes, where this condition is found, and these bacteria can live in those conditions. Fine. Okay. So this is all regarding the archaebacteria. Next we come to eubacteria. Eubacteria means true bacteria. Okay. So eubacteria, their cell wall is made up of peptidoglycan. Now this peptidoglycan consists of 2 things. First of all, sugars, N-acetyl-muramic acid and N-acetyl-glucosamine. And second is amino acids. That is why this peptidoglycan is also called as mucopeptide. Mucopeptide, fine? Okay. So this is the peptidoglycan. NAG and NAM are the 2 sugars, and then amino acids of different types. They make the cell wall of this bacteria. Fine. Now, some of them may be photosynthetic. These photosynthetic ones are called as the cyanobacteria. Cyanobacteria, they were earlier called as blue-green algae because earlier they were in the kingdom Plantae algae. Okay, they are in kingdom Plantae algae. Fine. Okay, so they were earlier, but now due to their prokaryotic nature, they have been placed in the eubacteria. So cyanobacteria are photosynthetic. Okay, for this they have photosynthetic pigment chlorophyll A. Remember, cyanobacteria are the only prokaryotes that contain chlorophyll A. There are other photosynthetic bacteria also like purple green bacteria, purple sulfur bacteria. They are also photosynthetic, but they do not contain chlorophyll A. They contain different pigments which are called as the bacterial chlorophyll. Fine. Next is chemoautotrophs. As I said, this is an exclusive mode of nutrition in bacteria where the organism is autotroph. It combines inorganic raw material to produce organic food. But one thing is there that they do not use sunlight. So they are not photoautotrophs. They will oxidize some inorganic compound like nitrites or nitrates. And the energy released in the process will be used for synthesis of food. They are called as chemoautotrophs, exclusive feature. Fine. So see here, chemoautotrophic bacteria oxidize various inorganic substances and such as nitrates, nitrites, ammonia, and use the released energy for their ATP production. They play a great role in the recycling of nutrients like nitrogen, phosphorus, and sulfur. But as I said, most of the bacteria are heterotrophic. Most of the bacteria are heterotrophic. Now, heterotrophic can be either saprophytic or parasitic, okay, fine, or symbiotic. So, saprophytic bacteria, they play important role in production of the curd. The bacteria that you use for production of curd, they are growing in dead organic matter. They are saprophytes. Bacteria that are used for production of antibiotics, okay, they are, for example, streptomyces, they are all saprophytic bacteria. Parasitic bacteria cause diseases. Okay. So they are pathogenic. For example, cholera, typhoid, tetanus. Citrus canker is a disease of lemon. Sometimes you see on lemon, you see brown spots. That is the bacterial disease called as citrus canker. Okay. Now, reproduction in bacteria. Children, remember, under favorable conditions, bacteria wants to increase its number as much as possible. Then it performs this process which is called as binary fission. Okay, so a mother cell will divide to form two daughter cells, and it is very rapidly they divide, and this is called as generation time. If you remember, in Escherichia coli it is just 20 minutes. Okay, within 20 minutes from one mother cell two daughter cells are formed, but a bacteria to bacteria it varies. Right? Okay, so under favorable condition it performs binary fission. But the conditions are unfavorable, then it produces different types of spores. And the most common is endospore. So remember two things, favorable condition binary fission, unfavorable condition spore formation. But genetic recombination in bacteria also takes place by a very primitive type of sexual reproduction. Remember, in bacterial sexual reproduction, there is no meiosis, there is no gamete formation, nothing like this. But a genetic recombination takes place. These are by three methods. One is called as conjugation. Another is called as transformation, okay, and third one is called as the transmission, okay. So transduction. So conjugation, transformation, transduction are the methods of genetic recombination in bacteria. It is a primitive type of sexual reproduction. Last thing is mycoplasma. They are the only wall-less bacteria. Mycoplasma do not contain cell wall. Second, these are the smallest living beings. Third, they are pathogenic also. And fourth, they can survive under anaerobic conditions also. Methanogens are also obligate anaerobes. Next, we come to kingdom Protista. All unicellular eukaryotes, exception with one or two are placed in kingdom Protista. So it is an artificial grouping. Members from other groups have been taken if they were unicellular eukaryotes in kingdom Protista. That is why the boundaries of kingdom Protista are not well defined. Same organism may be a plant to someone, a protist to another one. As I said, Chlamydomonas. Botanists claim that it should be the people plain that it should be a plant because it has chlorophyll, it has cellulosic cell wall. But other people say no, it is unicellular eukaryote, so it is a protist. So that is why boundary of this group is not defined. But they have a true nucleus. They have nuclear membranes present, so the true nucleus is present. Fine, okay. And they show sexual reproduction by cell fusion. They show asexual reproduction also. And Protista can be classified broadly into 3 types. Okay, so this is the basic classification. No, sulfur bacteria are not archaebacteria. Sulfur bacteria are eubacteria. No, methanogens are archaebacteria. Sulfur bacteria are eubacteria. Fine. Okay. So coming to photosynthetic protist chrysophytes, it includes desmides and diatoms. Now diatoms, the cell wall is made up of cellulose, but there is very heavy impregnation of silica in their cell wall. Diatoms are the chief producers of the ocean, first thing. Second, as I said, body is made up of 2 shells. So if you see diatoms, looks like a soap box where soap is kept. That is why they are also called a soap box organism. Third, the cell wall has heavy impregnation of silica. So even when they die, the cell wall is completely not destructed because of this silica. And this silica deposition keeps on continuing over millions of years. This silica deposition of the cell wall of the diatoms has made diatomaceous earth or diatomite, or it is called kiselgar. It is of economic use because it is porous and it is made up of silica. And you know, silica is chemically inert. So it is used for filtration of syrups and oils, etc. It is used for polishing. Okay, it is used for polishing because of its gritty nature. So that is about the diatoms. Coming to dinoflagellates. Dinoflagellates organisms, they have characterized by a transverse flagella and a posterior flagella. So these are dinoflagellates. They have plates of cellulose present on them. Okay, and they are of various colors depending upon the type of photosynthetic pigments in them. So brown, dark brown, right? Different colors they are present. Green, different colors on the photosynthetic pigment. And some of them, like goniolux, some of them, like goniolux, its number sometimes suddenly increase in the ocean. Many folds, millions of times increase. This is called as red tide. Okay, red tide. And they contain a toxin which is called as saxitoxin. Okay. This contain a toxin which is called as saxitoxin, which can kill other organisms of the sea, especially the other fishes. So let us see this 1-2 points very briefly. The diatoms have left behind large amount of cell wall deposits in the habitat. Accumulation over billion of years refers to diatomaceous earth being gritty. This soil is used for polishing, filtration, oil, syrup. Diatoms are the chief producers of the ocean. Fine. Next. They appear yellow, green, brown, blue, or depending upon their main pigment present in them. See, very often red dinoflagellates, goniolux, undergo such rapid multiplication that they make the sea appear red, called as red tide. Toxins released by such large number may kill other fishes. Last thing is euglenoids. Euglenoids are freshwater organisms. They have two flagella, one small flagella and one long flagella. So this is very characteristic feature, a small flagella and a long flagella, biflagellated. And they do not have cell wall. Remember, diatoms have cellulosic cell wall, dinoflagellates have cellulosic cell wall. Okay, but euglenoids do not have cell wall. The outer covering is called as pellicle. It is made up of flexible plates of protein, due to which it can show wriggling movement like this, contraction, expansion movement it can show. Fine, okay. And euglenoids, we have to remember one thing, that their photosynthetic pigments is similar to that of higher plant. That is, means they contain both chlorophyll A and chlorophyll B. They have two flagella short, though they are photosynthesis of sunlight. Okay, fine. Interestingly, the pigments of euglenoids are identical to those present in the higher plants. Second thing which makes euglenoid very, very important are mixotrophs. Generally, an organism is either autotrophic or heterotrophic, but euglenoids are mixotroph, means they are both autotrophs and heterotrophs because they have photosynthetic pigments. So when sunlight will be present, they will function as the photosynthetic autotrophs. But in absence of sunlight, they trap a small insect, then they become phagotrophs, means heterotrophs. So see here, though they are photosynthetic, in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating or other smaller organisms. Fine. So both autotroph and heterotroph. So with this, we have completed this photosynthetic protist. Now we come to slime molds. Slime molds, in their vegetative body, they have no cell wall. Okay. And under favorable condition, they keep on increasing their size, This is called as plasmodium. So under favorable condition, they produce plasmodium. And when conditions become unfavorable, they produce fruiting bodies. And these fruiting bodies, they produce spores, which are true spores. Why we are calling them as true spores? Because they have cellulosic cell wall. The spores have cellulosic cell wall. Chloroplasts are present in euglenoids. Yes. All photosynthetic eukaryotes have chloroplast. Remember one thing. Okay. All photosynthetic eukaryotes have chloroplast. Fine. Okay. So, slime molds, under unfavorable condition, they will produce fruiting bodies, and these fruiting bodies will produce spores which have cellulosic cell wall. So that is called as true spores. So remember, vegetative body without cell wall, but the spores have cell wall. And these spores are very resistant, and they survive for many years, and they are transported by air current. So this important small things we have to remember. The spores possess true wall. They are extremely resistant and survive for many years even under adverse condition. The spores are dispersed by air current. Fine. So this is all regarding the. Now we come to protozoan protist. Protozoan protists are of four types: amoeboid protozoans like amoeba, paramecium, etc. Okay, and they move. They do not have any flagella or cilia. They move by forming false food. Okay, which is called as the pseudopodia. Fine. They may be predator. Okay, they may be predators or they may be parasitic. Both are possible. For example, amoeba is a predator, but entamoeba is a parasite in our body, which causes the amoebic dysentery. Flagellated protozoans, trypanosoma. This is the reason for the cause of the sleeping sickness. Okay, so flagellated are pathogens. They cause sleeping sickness. Ciliated protozoans, example is paramecium, and cilia are very small hair-like outgrowths. Okay, and number is very high. So this from outside. Our normal proteins, they go unusual folding, abnormal folding, and convert into prion protein. But the problem is, if once a prion protein is formed, suppose folding is abnormal, if it comes in contact with a normal protein, it will convert it into a prion protein. So this is how prions multiply. A prion protein, when it comes under contact with a normal protein, it changes its folding pattern and converts into prion protein. And prions cause many animal human diseases. In animal, the most common disease is bovine spongiform encephalopathy, which is commonly called as mad cow disease. And the same disease in human beings is called as Creutzfeldt-Jakob disease. Okay, same disease is called as that is what I am saying. The latest NCRT, Vaishnavi, the latest NCRT that has come, now they have very clearly mentioned the name virus was given by W.M. Bejrik. Okay, but they keep on changing this. What can we do? Fine. Okay. Yes. So this is all regarding the prions, and human same disease is called as the Creutzfeldt-Jakob disease. Now, lichens are a symbiotic association between a fungi and an algae. That is why they are called as composite organisms. The fungal partner is called as mycobiont, and the algal partner is called as phycobiont. So together they are a myco-phycobiont. One of them is autotroph, that is algal partner is autotroph, and fungal partner is heterotroph. Algae has only one function in this symbiosis, that is photosynthesis, food production. All other functions are taken up by the fungal partner, that is water absorption, mineral absorption, shelter, structure, everything fungal partner. So this is a symbiotic association between an algal partner and a fungal partner. So children, with this we complete our second chapter, that is the biological classification, and we will move on to our third chapter, that is the plant kingdom. Fine. So plant kingdom, first of all, we will see classification. Artificial system, natural system, and phylogenetic system. When classification of plants is done only on one or few characters, okay, one or few characters, basically morphological characters or structure of the sex organs like stamen, endoecium, and gynoecium, then it is called as artificial system of classification. This classification system gives equal weightage to both reproductive and vegetative characters, and that is the biggest drawback of this system, because reproductive characters are more stable. Vegetative characters change with environment, so reproductive characters should be given more importance, but it gives equal importance. Second thing is, since criteria for classification is only morphology, very few, that is why many unrelated organisms come in the same group, and many related, they are separated. The most common is the sexual system of classification given by Carolus Linnaeus. Sexual system by Linnaeus, where he classified all plants on the basis of stamens. Okay, so this is artificial system. Natural system is based on natural affinity. So not only if two plants are same size tree, they will be put together. No, there should be natural closeness between them. So for our natural system of classification, all existing characters are studied. Morphology, anatomy, cell structure, everything, all existing characters are studied, but existing, not past. It does not take into account evolution. And on the basis of this, classification is done, which is called as natural system. And the most common is Bentham and Hooker's system. Bentham and Hooker system, natural system of classification. And phylogenetic is basically based on evolutionary relationship. So these are the three classification systems. Okay, most advanced is phylogenetic, but most practical uses of the natural system of classification. Now, in phylogenetic system of classification, is you see, if it is based on evolution, it is based on fossil records. And sometimes these fossil records are missing. When these fossil records are missing, then we take help of other things. That is the cytotaxonomy, where we try to classify on the basis of structure of cell, like the number and structure of stamens, number and structure of chromosomes. Then Numerical taxonomy. Numerical taxonomy is based on all observable characters. All characters are given equal weightage, okay, and numbers and codes are assigned to the characters, and classification is done on the basis of computer programmings. And chemotaxonomy. Chemotaxonomy. Chemotaxonomy means classification based on the chemicals produced by different plants. Okay, so there are, for example, there are many families where aromatic oils are produced. For example, Labiati family. Okay, so on the basis of which type of chemical producing, for example, you know there is a genus Rauwolfia. Different species of Rauwolfia produce a chemical called as reserpine. So this type of on the basis of the chemicals produced when classification is done, it is called as the chemotaxonomy. So these help in the phylogenetic system of classification where the fossil records are missing. Fine. Now, kingdom Plantae includes algae, Bryophyta, Pteridophyta, Gymnosperms, and Angiosperms. Okay, fine. So algae, Bryophyta, Gymnosperms, and Angiosperms, they are all included in the kingdom Plantae. Now coming to algae, you see main cheese which we have to remember in algae is this chart. Algae are mostly aquatic, maybe freshwater, marine water. They lack basically any tissue level of organization as such. Okay, very primitive structure. They do not form embryo, non-embryophyte plants. Okay, and main criteria of classification of algae is the photosynthetic pigments. Okay, main criteria photosynthetic pigment. But algae also differ in their reserve food and flagellation, type of flagella. So this is a chart which you have to remember for algae. Green algae, chlorophyceae. Major pigments, chlorophyll A, chlorophyll B. Stored food is starch, cell wall, cellulose, and flagella 2 to 8, apical and equal size. And this is freshwater, marine water, or brackish water. Brackish water means which is salinity is less than the sea water but more than the freshwater river. That is called as brackish water. Second is Phaeophyceae or brown algae. They have very, very huge complex body structures. Their photosynthetic pigments are chlorophyll A, chlorophyll C, and fucoxanthin. And the brown color comes due to this fucoxanthin, which is a type of a carotenoid. Stored food is mannitol and laminarin, cell walls, cellulose, but outer layer is of algin also. Flagellation, you can see 2 unequal size and apical flagella. Okay, sorry, lateral flagella, 2 unequal size, one long, one small. Mostly they are brown algae are mostly marine. Okay, mostly marine. Freshwater very rare. And third, red algae, chlorophyll A, chlorophyll D, and phycoerythrin. The red color comes due to the phycoerythrin. Stored food is Floridian starch, which is chemically more close to glycogen and amylopectin. Fine. Cellulose, inner cellulose, outer pectin, and polysulfate esters. These polysulfate esters can either be agar, as in Glaceraria and Gelidium, okay, or it can be carrageenan, okay. For example, porphyra, fine. Carrageenan, it can be carrageenan or it can be agar, fine. Next, there is no flagellation. This is very, very important point. No flagellated structures found in red algae, whether it is sexual spores or whether, gametes or it is asexual spores. Red algae do not produce any flagellated structures. They are also mostly marine, but they are freshwater forms also, fine. So this is basically you have to remember in algae. Now coming to Bryophyta. Bryophytes are more, the thallus structure is more advanced as compared to algae. And now we will see the alternation of generation. Remember children, in plants there are two stages in life cycle. The stage of the life that produces gametes, that is called as the gametophyte, and it is haploid. And the stage of life that produces spores by meiosis, sexual spores by meiosis, that stage is called as a sporophyte, that is diploid. And these two stages alternate with each other. Okay, these two stages alternate with each other, which is called as alternation of generation. Now, from bryophytes to angiosperms, generally, if we move from lower to higher, it is from predominant gametophyte to predominant sporophyte. So bryophyte's main plant body is the gametophyte, which produces the gametes. Okay, the sporophyte is completely dependent on the gametophyte throughout its life. So this is a characteristic feature of bryophytes, that the sporophyte dependent on the gametophyte. Okay, bryophytes are the first land plants. First plants to produce the female sex organ Archegonia. Before that it was Oogonia. Okay, and they are called as the amphibians of the plant kingdom, because although they have come on the land, but they require water for their fertilization. That is why their distribution is not very wide. Okay, now bryophytes are mainly divided into two classes, that is the liverworts or Hepaticopsida and the mosses or the Bryopsida. Okay, fine. So liverworts, they are primitive as compared to the mosses. Liverworts, they have, for attachment on the surface, they have hair-like structures called as rhizoids. The rhizoids are unicellular, and member of liverworts are Rixia and Markensia. Markensia is a dioecious plant. See here, Markensia is a dioecious plant. Male and female thallus are completely different from each other. Okay, male and female thallus. The female thallus, it produces an archegoniophore which bears archegonia. The male thallus produces an anthridiophore which produces the anthridia. Okay, no other bryophyte you will find which shows this dioecious condition. Only Markensia is an exception which shows dioecious condition. Fine. Okay, all other bryophytes are monoecious, means both anthridia and archegonia are produced on the same plant. Fine. Now coming to mosses. In mosses there is a third stage in between. Fine. Okay, I told you mosses. The gametophyte has two stages. First is called as protonema stage. This is a characteristic feature of mosses, please remember, which is the juvenile or early stage, branched, green, filamentous structure. Okay, fine. And then the leafy stage. Adult has axis that bears leaves. Fine. So this gametophyte having a protonema stage, this is a characteristic feature of mosses not found in the liverworts. And the plant body of mosses is more advanced as compared to the liverworts. Fine. Plant body of mosses are more advanced as compared to the liverworts. Now, yes, bryophytes vascular tissue absent. Vascular tissue start from the, remember children, it is a marathon class, so each and every line cannot be explained. Okay, for that will take at least 25 hours. So basic things can only be explained because you have to cover entire 11th chapter. Okay, fine. Yes, so bryophytes are of very less economic importance. One basic economic importance, two are there, and same bryophyte, that is the sphagnum. There is a plant called as sphagnum. This plant, sphagnum, grows in acidic bogs. So when the plant dies, it is not completely decomposed because of this condition. So it keeps on accumulation, and this accumulation ultimately turns into a compressed coal-like structure called as peat, which is used as a fuel. Okay, which is used as a fuel. Fine. So first of all, it is the source of peat, which is fuel. Second, the sphagnum can store much more water than its own weight. That is why it is used as a packaging material for transshipment of living tissues. Fine. So these are the two economic importance. But ecological importance is there. Ecological importance is that along with the lichens, they are the pioneers of the succession on a rock. Fine. And second, they are very good soil binders. So they decrease the impact of the falling raindrop and prevent soil erosion. That is the ecological importance. Fine. So this is all regarding the bryophytes. Then we come to pteridophytes. So these are the first vascular land plants. Bryophytes were first land plant. These are the first vascular land plants. Okay. Of course, pteridophytes are more advanced as compared to the bryophytes. And here the main plant body is sporophyte. And the spores are produced inside the structures called as sporangia. If we draw the life cycle, it will come like this. Okay, this is life cycle. So see, this is diploid, this is diploid, this is diploid, but after meiosis, this is haploid, this is haploid, 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 this is diploid, again this is diploid. So if we draw a line, the stage of life below the line, you see, this is called as the gametophytic phase. And above the line, it is called as sporophytic phase. Fine. So there is alternation of gametophyte and sporophyte. There is alternation of gametophyte and sporophyte in the life cycle. Okay. Now in bryophytes, the gametophyte is very minute but independent structure. It is photosynthetic structure, and this is called as prothallus. So remember, the gametophyte of the pteridophytes is collectively called as prothallus. It is free living. Remember, it is free living. Pteridophytes, both sporophyte and gametophyte, are free living. But main plant is the vascular sporophyte, and gametophyte is very minute, microscopic, small structure and very short-lived, which is called as the prothallus. Okay. So protonema is the gametophyte of mosses. Prothallus is the gametophyte of pteridophytes. Okay. Remember this point. Okay. Now pteridophytes, they are grouped into Psilopsida, Lycopsida, Sphenopsida, and Pteropsida. all members of Pteropsida are called as ferns. So don't think that all pteridophytes are ferns. Only these are called as ferns. Here example is Xylotum. Here example are Lycopodium and Selaginella. Here example is Equisetum. Here example is Adiantum, Dryopteris, Salvinia, Azolla. Fine. Now remember that most of the pteridophytes produce only one type of a spore. Most of the pteridophytes produce only one type of a spore. That is why they are called as homosporous. But some pteridophytes produce two types of spores. Large spores and small spores. This is called as heterosporous. Here example are Selaginella and Salvinia. This is of great evolutionary significance. That is why this was the first step towards seed formation. Although pteridophytes don't produce seed, but the first step towards seed formation. And later on, when the next group came, Gymnosperm, then seed formation started. Okay, this is the heterospory. Fine. Next, we come to Gymnosperms. Gymnos means plants. Gymnos means naked, and sperma means seed. Means these are naked seed plant. Means plants that produce naked seed. Means Salvinia. Well, I have written here Salvinia. I have written here Salvinia. See, Pteropsida. That is the ferns. Okay. Haji. So plant that produce naked seed. So seed formation started. Up till pteridophyte, no seed formation. So plant that do not produce seed are called as cryptogams. So algae, bryophytes and pteridophytes are cryptogams. Gymnosperms and angiosperms that produce seed are called as phanerogams. Okay. Okay. So this is the naked seed plant. Here also the main plant is the sporophyte, which is a very robust, very tall trees. Generally, you see pinus, citrus, very tall trees. Some are shrubs. Herbs are generally not found here. Shrubs, but mostly they are very tall trees. And one of the tallest gymnosperm is sequoia, which is called as the redwood tree. Okay, which is called as the redwood tree. Fine. If you see in gymnosperms also, the main plant is a sporophyte. Okay, main plant is sporophyte. It produces cones. And in cone there are sporangia. And in sporangia there are production of spores. So main plant contains cones. Cones have sporangia, and in sporangia there are production of spores. But all gymnosperms are heterosporous. All are heterosporous. Okay. So on the basis of this, male cone produces microspores. Because these microspores, they germinate to form the male gametophyte. And female cone produces megaspores. Because the megaspores germinate to form the female gametophyte. So there are separate male and female cones. Now, if both are produced on the same plant, then the plant is called as monoecious. For example, in pinus, male cone and female cone both are produced on the same plant. So pinus is a monoecious plant. But in cycas, there are two types of plants. On one plant, only the megasporophylls are produced, okay, and which produce only the megaspores. And on one plant, only the male cones are produced. So that is why cycas is called as dioecious, because the male and female plants are distinct in the cycas. Fine. Okay. Next, coming to life cycle, let us try to see what happens. The plant will produce cones, and on cones there will be spore mother cells. Spore mother cells will undergo meiosis. I will draw the complete diagram so you understand. The megaspore is called as ovule. Megasporangia is called as ovule and microsporangia. In megasporangia, there is megaspore mother cell. In microsporangia, there is microspore mother cell. This undergoes meiosis. And this undergoes meiosis. This will produce megaspore. And this will produce microspore. Megaspore will produce female gametophyte. And microspore will produce male gametophyte or sperm. Or pollen grain. Fine. Then this pollen grain, by pollination, will be transferred to the female gametophyte. Okay? And there will be fertilization to form the zygote. From zygote to embryo, and embryo will give rise to the main plant. So here also see, this is the sporophytic phase. This is gametophytic phase. Of life cycle, okay? Fine. So this is the life cycle of the gymnosperms, and gymnosperms are much more advanced as compared to the pteridophytes because they start producing seed while pteridophytes do not produce seeds. Fine. Okay. So this is all regarding the life cycle of gymnosperms. Fine. So with this, children, we have completed kingdom Plantae. So 3 parts we have completed. That is the living world, biological classification, and plant kingdom. Few questions are there. We will see in brief terms 5, 6, 7, 8, 10 questions, and then we'll move on to the further. Let us see. Now, reproduction is synonymous with growth in which of the following organism? So I told you that in unicellular organism, we cannot differentiate between growth and reproduction. So it means you have to find unicellular organism. So here you see bacteria, unicellular algae, and amoeba. This will be our answer because fungi is multicellular. Here fungi is multicellular and filamentous algae are multicellular. So answer will be first. Next. Select the correct written scientific name of mango, which was the first discovered by Carolus Linnaeus. So I said first name, generic name should be capital, specific name should be small, and the name of author but not italics. So you see, first one is correct. Second one, because author name is missing. Third one is incorrect because specific name is capital. Okay, and here because complete name is written Carolus Linnaeus, that is why this is wrong. Fine. Okay, next.
Higher taxa share, we have said that higher taxa share lesser common characters. Higher the taxa, lesser the common characters. That is why classification becomes very, very difficult. Which one of the following statement has organisms scientifically correctly named and correctly printed? See, Musca domestica, the common lizard, wrong, because Musca domestica is the housefly. Plasmodium falciparum, a protozoan pathogen causing malaria. This is correct. Tigris is the Indian tiger. Gir forest. No, Gir forest is famous for Asiatic lions, not tiger. And E. coli full name is Entamoeba coli, a commonly occurring. No. Entamoeba is a protozoan. It should be Escherichia coli. So this is wrong. So answer will be B. Plasmodium falciparum. See. Next. Match this, leave this one. Such match questions also come where you have to label the diagram. So see here, it is A is the nostoc, B is heterocyst, and C is the mucilaginous sheath. So nostoc, heterocyst, and mucilaginous sheath diagram question is there. Chrysophytes, euglenoid, dinoflagellates, and slime molds are included in kingdom Protista. Not very difficult, but it has been asked in Neet, that is why. Oxygen is not produced during photosynthesis. Remember that purple sulfur bacteria, green sulfur bacteria, they do not produce oxygen during photosynthesis. Fine? Okay. This question, 2017 question, let us do. Which among the following are the smallest living cells known? Only first line will tell, it should be mycoplasma, okay? The smallest living cell, mycoplasma. Which of the following is the correct sequence of class mycelium fruiting body observed in fungi? Phycomycetes septate, wrong, because Phycomycetes have cenocytic. Ascomycetes aseptate, again wrong. Epsomycetes have septate. Basidiomycetes aseptate, again wrong, because Basidiomycetes have septate. Deuteromycetes septate and branched, and fruiting body is not present. Very good, because they do not show sexual reproduction, or sexual reproduction is absent. So answer will be D. Deuteromycetes, septate mycelium, and fruiting body is not present. Such labeling may come, please see. Okay, this is head, this is called as collar, and this is the tail sheath, and this are called as the tail fibers. So please see these labelings. See one assertion reason question. Two kingdom classification used for a long time was found to be inadequate. Why? The two kingdom system of classification did not distinguish between eukaryotes and prokaryotes, unicellular and multicellular, and green algae and fungi. It means reason is the correct explanation of assertion. Both assertion and reason are correct, and reason is the correct explanation of assertion. Fine? See one more. Chemoautotrophic bacteria are nitrifying bacteria, A1, correct. Photoautotrophic bacteria, purple and green bacteria, 2 and B2 also correct. Symbiotic bacteria, yes, Rhizobium and Frankia, they perform nitrogen fixation, this also correct. And parasitic bacteria, this causes cholera. So A, B, C, D, 1, 2, 3, 4. So answer is 1, okay? Answer is 1, 1, 2, 3, 4, all same sequence, it is correct. This is very, very important. Okay. And fill up the blanks. You have to fill the blanks here. Chlorophyce, major pigment will be chlorophyll A and B. B. Okay. Here, cell wall, phaeophyce, cellulose and algin. Okay. Here, stored food, rhizophyce, Floridian starch. Floridian starch. Okay. And that is A, B, D. Rhodophyce, That is, they fresh water also, brackish water also, and marine also. All three. So this way we can fill this blanks. Fine here. Which of the wrongly matched? Gyma cup marschensia, yes, correct, because these are the asexual structures. Biflagellated zoospores, brown algae, this is also correct. Uniflagellated gametes, polysiphonia, no. Okay, polysiphonia, Porphyra, polysiphonia, they are called as the red algae and their flagella is completely absent. And unicellular organisms, chlorella, correct. So our answer will be C. Polysiphonia does not produce uniflagellated gametes, so answer will be C. Ajay, select the correct sequence arrangement of reproductive structures in pteridophytes. Strobili will come first. Cone of strobili, sporophyll, sporangia, spore. This is the correct sequence. Strobilus, then sporophyll. Within on the sporophyll is sporangia, and within sporangia spores. So answer is B. Fine. One last question we are going to do. Last one. This we can do. Assertion reason. See. Artificial system of classification separated closely related species. Yes, because it was only on one criteria. This is a correct statement. Artificial system gave equal weightage to vegetative and sexual. Yeah, this is correct. Statement 1 is also correct. Okay. Statement 2 is also correct. Okay. But reason is not the correct explanation. Reason is not the correct explanation. Okay. Because this is related to stability and this is related to the criteria. So both are correct, but reason is not the correct explanation of the assertion. So children, with this, a 4-5 minutes break we will take, and we will then start with the cell, the unit of life. Fine. 4-5 minutes in between, you can have water or some snacks you have, you can have it. Okay. 4-5 minutes, hardly 5 minutes break, and then we will start again. Okay. Fine. ये मैं खा लिया। क्या है? चलो ठीक है, थैंक यू। नहीं नहीं नहीं,
बनाना सर, बनाना।
Okay, let's just resume. Are you back? Okay. Ajay, so we are starting cell, the unit of life. Okay. But as we say that nowadays it is more appropriate to say cell phones, the unit of life. But anyway, cell, the unit of life. Fine. Okay. You see, the basic unit, suppose our body, what is the basic building block of our body? That is a cell. Metabolism, genetics, everything is taking in place the cell. Then the cell to tissue, tissue to organ system. But if we keep on separating, ultimately we will get cell. So cell is the basic structural as well as functional unit of life. Fine? Okay. Now, if we see cell, basically cell theory, that Sheldon and Schwann, two scientists, they studied that. Sheldon studied plant cells. He was a botany, he studied plant cell, and he said all living beings are made up of cells. Then there was another scientist, he was a zoologist, that Schwann, he studied plant cells also and animal cells also, and he said body of plants and animals are made up of cells. And he said that apart from that, the plant cell has a cell wall also, which is not present in the animal cell. So Sheldon and Schwann together proposed the cell theory, which they said that plants and animals are made up of cells. Fine. But they could not tell how new cells arise. This was explained by another scientist, which is called as Rudolf Virchow. Okay, Rudolf Virchow gave omnicellular e-cellular, means new cells arise from pre-existing cells, already present cells. Pre-existing means already present cell. So he gave the modified cell theory where two criteria, 2 points were there. First, that body of plants and animals are made up of cells, and second, new cells arise by cell division in the pre-existing cells. Now, if you see an overall view of the cell, so a cell has an outer membranous structure which is called as a cell membrane. The entire material present within the cell membrane is called as the protoplasm. Okay, the protoplasm has two parts. Membrane plus protoplasm is equal to protoplast. Okay, excluding membrane, it is protoplasm. Now, protoplasm has two parts, a nucleus and cytoplasm. Now, on this basis of nucleus, there are two types of cells. If a nuclear membrane is absent, true nucleus is absent, then it is called as prokaryotic cell like bacteria. And if a nuclear membrane is present, true nucleus is present, then it is called as eukaryotic cell like plants, animals, all of us. Fine. Okay, except Monera, all are eukaryotes. Next. Cytoplasm is the main arena of the cellular activity. Remember, because all metabolic processes take place in cytoplasm. Again, on the basis of cytoplasm, we can see difference between the eukaryotic cell and prokaryotic cell. Okay, prokaryotic cell do not have membrane-bound organelles in cytoplasm, while eukaryotic cell have many membrane-bound organelles in the cytoplasm, like chloroplast, mitochondria, endoplasmic reticulum, Golgi body, lysosomes, many such are present. Okay, and ribosomes are present in both. Ribosomes are also organelles, but are present in both.
These living components are called as organelles. Now, ribosome is a naked organelle. It is not membrane-bound organelle. It is present in both prokaryotic and eukaryotic cell. Difference is in prokaryotic cell, it is smaller and lower density called as 70S. In eukaryotes, it is larger and higher density called as 80S ribosome. Okay. Apart from that, one more naked organelle is present in eukaryotic cell, but only in animal cell, which is called as the centrosome. Called the centrosome. Fine. So this is all the overview of the cell. Now, say shape, size all of the cell, it will depend upon the function it has to perform where it is present. For example, nerve cell have to conduct the nerve impulse, so that they are very long cells. Okay. Now the columnar cells of the intestine, they are for absorption, so they have microvilli. So depending upon what function has to be performed, the shape and size of the cell varies. For example, human RBC, as it matures, it becomes enucleated, no nucleus is not present, and it is spherical but biconcave. Okay, biconcave, fine. Okay. So first of all, we are going to do the prokaryotic cell. So prokaryotic cell, let us see. We have already said that all prokaryotes are collectively called as bacteria. All prokaryotes are collectively called as bacteria. So this is a bacterial cell, you can see here. Now, three things are there: capsule, cell wall, and plasma membrane. All three are together called as cell envelope. Okay. Now, as far as capsule are concerned, basically it is called as glycocalyx. It is made up of sugars. If it is hard, then it is called as capsule. Hard and thick, then capsule, and if it is thin, then it is called as slime layer. Fine. Now, cell wall, coming to cell wall. Cell wall is made up of peptidoglycan, that I have already told you, that peptidoglycan is made up of two types of sugars, NAG and NAM, and different types of amino acids. Okay, L-alanine, D-alanine, and different types of amino acids. Okay. Cell wall gives shape to the cell. Okay, give shape to the cell, provide rigidity and mechanical support to the cell. Okay, and they protect the cell from mechanical injury, etc. Fine. And plasma membrane, this is the only structure. Remember, children, very important. This is the only structure of prokaryotic cell or bacterial cell which is similar to the eukaryotic cell. All other things are different, but the structure of the plasma membrane of prokaryotic cell and eukaryotic cell is similar. It is a common feature. Fine. Okay. Now, cell envelope. Okay. Now, plasma membrane, at certain place, it shows infolding called as mesosomes. So this infolding of plasma membrane is called as mesosomes. Mesosomes have 4 or 5 very important functions. First, in aerobic bacteria, they contains the enzymes of the electron transport system and oxidative phosphorylation. So in this way, they performing the function of the mitochondria in prokaryotic cell. So if it is aerobic bacteria, then the function of mitochondria, 2 functions, that is the electron transport system and oxidative phosphorylation, ATP synthesis is performed on the mesosome. Second, mesosomes help in cell division, binary fission. Okay. Third, mesosomes help in DNA replication. Okay, mesosomes help in DNA replication. So there are so many functions of the mesosome associated with the bacterial cell. Fine. Next, ribosomes. They are smaller in size, called as the 70S ribosomes, which have a 30S smaller subunit and a 50S larger subunit. Sometimes many ribosomes get attached to the same mRNA. Sometimes many ribosomes get attached to the same mRNA, and this structure is called as polysome. So cytoribosome we have inclusion bodies. Children, this is in Inclusion bodies, this is unique feature of prokaryotes. Okay. Inclusion bodies are not found in eukaryotic cell. Inclusion bodies, the question has been asked earlier, they are non-living, non-membrane bound structures. They are basically stored food in the form of synophycean granule or phosphate granule or volutin granule, or they may be gas vacuoles. Gas vacuoles are found in purple and green bacteria. These come under inclusion bodies. They are non-living structures. Next, nucleoid, very important. In bacteria, there is no nuclear membrane. Prokaryotic cell, no nuclear membrane. So this primitive type of nucleus is called as nucleoid. It contains naked single molecule of circular double-stranded DNA. Again, listen carefully. I'm not saying single-stranded DNA. I'm saying single molecule of circular double-stranded DNA not associated with histone proteins to form chromatin. Chromatin is found only in eukaryotic cell where the DNA is associated with histone to form chromatin. No chromatin is formed in prokaryotic cell because there is DNA is not associated with histone protein. So this is called as nucleoid. Okay, fine. Plasmid. Certain bacteria, not all, certain bacteria contain some additional DNA also. These are called as extra chromosomal DNA or extra nuclear DNA. These are called as plasmid. These are also circular double-stranded DNA which are useful but not essential for the survival of bacteria. If they are present, they confer some additional abilities like antibiotic resistance that you have studied in biotechnology, resistance against antibiotics, disease causing ability of the Agrobacterium tumefaciens is due to a plasmid called as TI plasmid. In many Escherichia coli, it may provide conjugation. Okay, so many such things are additional genes are present. In rhizobium, the genes for nitrogen fixation are present on the plasmid. So these are extra chromosomal circular double-stranded DNA. But interesting thing is that they have their own origin of replication. So they are autonomously replicating or self-replicating. That is why plasmids are used as a cloning vector in the biotechnology due to their self-replicating ability or presence of ORI. Next, Pili are small bristle-like structures present on the entire surface. Okay, pili are small bristle-like structures present on the entire surface of bacteria. They help in attachment to a substratum or attachment to the host. Fine. Flagella. The flagella are locomotory structures of bacteria. Okay, they are not exactly similar to the eukaryotic flagella. Bacterial flagella has a basal body and a hook and a filament. Hook-like structure is not found in eukaryotic flagella. A basal body, this is basal body, this is hook, and this is filament. Filament does not have the typical 9+2 arrangement. Okay, it is made up of flagellin protein. Okay, it is made up of flagellin protein. Okay, and this pilus, it is made up of special proteins called as the pilin protein. Apart from this, okay, there are some bristle-like structures which are called as fimbriae. In fact, this fimbriae help in the attachment to the substratum, and these are bristle-like structures. This pilus, basically pilin, it is made up of pilin protein. It helps in the conjugation of the bacteria. Okay, so pilus are larger than the fimbriae but smaller than the flagella. But locomotory structures are only flagella. Pilus and fimbriae do not help in locomotion. So this is a simple basic structure of the prokaryotic cell. Next we come to eukaryotic cell. Eukaryotic cell wall,
only in fungi and plants. You can say Protista also, they are photosynthetic protist. Okay. And protoplast. Okay. Now this protoplast, cell membrane and protoplasm. So those who are confused that, well, sir, what is the difference between protoplast and protoplasm? I think now your confusion is clear. What is the difference between protoplast and protoplasm? Is it? Are you there? Now, this protoplasm is divided into two things: cytoplasm and nucleus. Okay. Okay. This cytoplasm is further divided into two parts mainly, that is cytosol. The fluid, clear fluid, is called a cytosol, and organelles. These are the living components, okay? And nucleus is divided into nuclear envelope, nucleoplasm, and in the nucleoplasm there are two things: nucleolus and chromatin. Okay. Organelles can be naked, ribosome, and centrosome only in animal cell, and membrane bound. Endoplasmic reticulum, Golgi complex, mitochondria, lysosome, plastids, microbodies, these are the membrane bound organelles. So this is an overall view of the cell. Fine. Is this chart clear to everyone? So we will be starting with the cell wall. Okay, we will be starting with the cell wall. Let us see. Now see, cell wall gives protection
mechanical damage, helps in cell-to-cell interaction, and provides barrier to undesirable macromolecules. Now, algal cell wall is made up of cellulose and galactans and mannans and minerals like calcium, while in other plants consist of cellulose, hemicellulose, pectin, and protease. So remember this chemical composition you have to remember. Okay, this chemical composition you have to remember. The cell wall, you see, if you see a cell wall, this is the primary wall, which is formed first. And inner to the primary wall is formed the secondary wall. So suppose this is the primary wall, inner to the primary wall is formed the secondary wall. Till only primary wall is present, primary wall is flexible. So cell will grow, okay, keep on growing, okay. But once secondary wall deposition takes place, growth of the cell stops and gradually the primary wall diminishes, okay, then secondary wall is only present, fine. Okay, next, structures associated to the cell wall, two things we have to remember. Okay, between two cells, there is a cementing material which is called as middle lamella, which is made up of calcium pectate. So the adjacent plant cells are connected to each other through middle lamella, which is made up of calcium pectate, and the cell wall is middle lamella, maybe traversed, and between the two cells, sometimes there are pores. Both cell wall and middle lamella, they have pores through which there is movement of substances from cell to cell. These are called as plasmodesmata. So these plasmodesmata are cytoplasmic bridge between adjacent plant cells. So remember this plasmodesmata, question is asked, cell wall, question is asked. Fine. Now coming to the cell membrane. Yes. Cell membrane is a quasi-fluid structure, means not completely hard solid, but not also completely water. Partially fluid structure. Okay. And cell membrane has two major components, that is phospholipids or phosphoglycerides and protein. Apart from that, it also contains a small amount of carbohydrates. Okay. So what is the arrangement here? See, blue ones you see, these are the phospholipids. So phospholipids form bilayer in the cell membrane. Okay. And in both the layers, the head portion is towards the outer side and the tail is towards inner side. The head is hydrophilic, okay, and polar, and the tail is hydrophobic and non-polar. Fine. So this arrangement is such so that the tail directly does not come in contact with water because, as I said, tail is hydrophobic. So to protect the cell from the water, in both the layers, the tail, hydrophobic tail, is towards the inner side. Okay. And this phospholipid can very easily laterally move within the cell. And the proteins are embedded in the lipid bilayer. You see, these are the proteins. As a result, proteins can also laterally move. How easily the proteins can move within the lipid bilayer, this determines the fluidity of the membrane. More easily they can move, more fluid is the membrane. Less easily they can move, less fluid is the membrane. So this is phospholipid. Apart from phospholipid, cell membrane also has one more lipid which is called as cholesterol. This cholesterol, this regulates the fluidity of the membrane. It prevents too much fluidity and it also prevents too much rigidity. So it regulates the fluidity of the membrane, cholesterol. Second is proteins. Proteins are of two types. Some are present only on the surface. So these surface proteins are called as extrinsic proteins or external proteins, and they can be very easily separated. Okay. Extrinsic proteins or peripheral proteins present only on the surface and they can be easily separated by using detergent. While there are other proteins which are called as intrinsic proteins or integral proteins, they are embedded, partially or completely embedded in the membrane. They cannot be easily separated and they are called as integral proteins. So depending upon the ease of separation, they can be peripheral proteins or integral proteins. Now, coming to the third component, that is sugar. Sugar is present only toward the outer surface. You see, no sugar is present toward the inner surface. And this arrangement of sugar makes the membrane, the two faces of the membrane asymmetrical. The two faces are asymmetrical because sugars are present only on the outer surface. Fine. Okay. Now, children, the sugar can either be attached to the protein. Okay. These sugars. Okay. Then what you will call it? Glycoprotein.
or they can be attached to the lipid, then you will call it glycolipid. That is why NCERT has shown different colors. So yellow one you see attached to protein, this is glycoprotein, and the green one attached to the lipid, this is called as the glycolipid. Now, fluidity of the membrane is necessary. Why fluidity of the membrane is necessary? Let us try to see. Because fluidity makes the membrane flexible. And this fluidity of nature of membrane is essential for so many functions. See, growth, formation of intercellular junctions, secretion, endocytosis, and cell division. Okay. Now coming to the membrane transport. If ATP is not used, energy is not used, then the substance will move from the region of their higher concentration toward the region of their lower concentration. This is called as the passive transport. Okay, passive transport. Okay, spontaneous movement. This passive transport can be of two types. If membrane proteins are not involved in passive transport, they are directly moving through the lipid bilayer. It is called as simple diffusion. And if membrane proteins, through membrane protein, this passive transport is taking place, then it is called as facilitated diffusion. Fine. Now, polar molecules cannot pass through the lipid bilayer, which means they cannot perform simple diffusion. So, polar molecules. Neutral solutes can move lipid bilayer simple diffusion, but polar charge always move through the membrane protein. So, they show facilitated diffusion. Opposite to that, if things have to be moved from lower to higher concentration, means uphill transport, then always energy will be used. That is why this transport is called as active transport. And active transport is always through the membrane proteins, and these membrane proteins are called as pumps. For example, sodium potassium pump. Fine, sodium potassium pump. So this is all regarding the cell membrane. Cell wall we have already done. Now children, there are three organelles which work in very close coordination. These three organelles are membrane bound organelles, all three, and they work in close coordination. That is why they are called as the endomembrane system. Let us try to see how. Suppose this is RER. Suppose there is a rough endoplasmic reticulum. Suppose it produces a protein. This protein will be packed in a membrane, and this is called as transition vesicle. This transition vesicle will go and fuse with the Golgi complex. This phase of Golgi complex is called as the cis or forming phase. And as this protein will move from here to here, the non-protein part, remember most of the enzymes also have a non-protein part which is called as the apophag enzyme. Okay, so non-protein part will be added to it. It will convert into an active enzyme, and from there it will be released as a secretory vesicles. And some secretory vesicles form lysosomes. That is why lysosomes are a storehouse of enzymes. So see, normal simple protein produced by the rough endoplasmic reticulum, transported to cis phase of the Golgi complex. From when it moves from cis phase to transverse, it get complexed, elaborated. It attached the non-protein part and converted it to enzyme. And then it produced, released from the trans phase as secretory vesicles, and some of the secretory vesicles form lysosome. It means rough endoplasmic reticulum, Golgi complex, and lysosomes, they are working in close coordination with each other. That is why they are part of the endomembrane system. And if you see a plant cell, you can add one more thing, and that is vacuole. So these four are the part of the endomembrane system. I just want you to tell me whether you have understood it, why it is called as endomembrane system, why not all membrane bound organelles are part of the endomembrane system. Is it clear? Okay, fine.
Thank you. So let us see each one by one. Endoplasmic reticulum, I told you, endoplasmic reticulum, it forms a membranous network in the cell which divide the cell into two parts. Okay, thank you. It divides it into two parts. The space surrounded by the AR is called as a luminal space, and space outside is called as extraluminal space, right? Now, endoplasmic reticulum are of two types. Some of them have ribosomes present on their surface. These are the 80S ribosomes, remember. Those that have ribosomes present on their surface, they are called as the rough endoplasmic reticulum, and these are mainly involved in protein synthesis. So cell that is actively involved in protein synthesis have rough endoplasmic reticulum. On the other hand, if you see this one, they have no ribosomes attached on their surface. These are called as the smooth endoplasmic reticulum, and they are the site for the synthesis of lipid and steroidal hormones. You know, steroidal hormones are also lipids. So a cell that is involved in lipid synthesis and secretion will have large amount of this SER. And also in animals where there is production of the steroidal hormones, there will be large amount of smooth endoplasmic reticulum. So this is endoplasmic reticulum. Next you come to Golgi complex. I already told you Golgi complex consists of structures called as cisternae. Okay. Now the cisternae are arranged in such a way that there is a convex face which is called as the cis phase or forming phase, and there is a concave phase which is called as the trans phase or maturing phase. Okay, trans phase or maturing phase. Okay, and Golgi complex has 2-3 functions. Packaging of materials. The material that was sent by RER, it was complexed and it was packaged to be delivered either to the intracellular stage or secreted outside. Materials to be packaged in the form of vesicles. These are called as the secretory vesicles. From the ER, fuse with the cis, what I have just told you, let's see, phase of the Golgi apparatus and move towards the maturing phase. This explains why Golgi apparatus remains in close association with the endoplasmic reticulum. Okay, fine. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in this Golgi apparatus before they are released from its trans phase. Protein produced by ER, converted into enzyme by Golgi complex, released as secretory vesicles, and secretory vesicles form the lysosomes. That is why we say the lysosomes are produced by Golgi body. Golgi apparatus is the formation of glycoproteins and glycolipids. Fine. Next, lysosome. See the first line in lysosome, what is written it? They are membrane-bound vesicle structure formed by the process of packaging in the Golgi apparatus. Okay, and lysosomes, they are the storehouse of various digestive enzymes, proteases, carbohydrates, lipases, nucleases. But one thing is that all these enzymes work at a pH that is acidic, that is less than 7 pH they work. Okay, so that is why these enzymes are called as acid hydrolases, because all digestive enzymes, they break the bonds by using water. That is why it is called as hydrolysis, whether it is digestion of protein or carbohydrate or nucleic acids or lipid. The bonds are broken by using water. That is why it is called as hydrolysis. And enzymes are hydrolases. And since they work on pH below 7, they are called as acid hydrolases. Fine. Okay. Next. Vacuoles. These are membrane-bound spaces. And the space or the membrane of the vacuole is called as tonoplast. Now, vacuoles are of various types. Here we are talking mainly about the vacuoles present in plants which are called as sap vacuoles. Okay, so they contain excretory material, water, many things, etc. Okay, and their concentration, solute concentration, is higher than that of the cytoplasm. Fine. As the plant cell becomes old, more and more materials are deposited in the vacuoles. As a result, the size of the vacuole keeps on increasing, so much so that in a old plant cell, 90% or more than 90% of the space is occupied by the vacuoles. Fine. Second thing is, children, which is called as the food vacuoles. Food vacuoles are vacuoles that contain food. These will be found in organisms like the protozoans. Okay.
that engulf the food and form vacuoles. These are called as the food vacuoles. Third one is called as the contractile vacuoles. These vacuoles are found in protozoans that live in water. So to remove excess water, to remove excretory product. So for osmoregulation and excretion, the vacuoles which are present are called as the contractile vacuoles. So remember, eukaryotes have three types of vacuoles: sap vacuoles, contractile vacuoles, and the food vacuoles. Prokaryotes have also a vacuole which contains metabolic gases, that is called as the gas vacuole. Okay, especially in the purple and green bacteria. Fine. So remember, prokaryotes have gas vacuoles, while eukaryotes have sap vacuoles, contractile vacuoles, and food vacuoles. Okay. Now, mitochondria is a separate organelle. Okay, it is not a part of the endomembrane system. Mitochondria are double membrane bound structures. You can see these all diagrams are, children, also very, very important. Mitochondria are double membrane bound structures. Okay. Outer membrane is continuous. It does not show any infolding. Inner membrane has many infoldings called as cristae. Okay. And due to these two membranes, the mitochondria has two chambers. The outer chamber, which is present between the outer and the inner membrane, and the inner chamber, which is surrounded by the inner membrane. Okay, fine. The inner chamber is called as the matrix, which contains the enzymes of the respiration, aerobic respiration, that is the link reaction and the Krebs cycle. Okay, and the inner membrane, the infolding is present, which contains the component of electron transport system. That is why a major function of mitochondria is aerobic respiration to produce ATP. That is why mitochondria is called as the energy currency of the cell. Apart from this, mitochondria also has the ability to synthesize certain of its own proteins, because the matrix also contains circular double-stranded DNA. Again, I'm saying double-stranded, remember, circular double-stranded DNA and 70S ribosome. So with the help of DNA, they can perform transcription to produce mRNA, and with the help of ribosome, they can perform translation to produce protein. So some of the proteins mitochondria is able to produce. That is why mitochondria is called as a semi-autonomous organelle, means it is partially independent of the nuclear genes because it has its own genes. Fine. Okay. This is mitochondria. Next we come to plastids. Now plastids are very large organelles. Mitochondria, normally you cannot see mitochondria, although it is very large organelle. So we have to give a special stain, genus gain B, and then we are able to see the mitochondria under the microscope after special staining. Now plastids are present only in the plants and euglenoids. Plastids, fine. Plastids are also double membrane bound structures. Now plastids can be classified into three types. So let us see. Okay. Now, green plastids, they contain both chlorophyll A and chlorophyll B, and also contain carotenoids. Okay. So since they contain both chlorophyll and carotenoid, but the predominant color is of the chlorophyll, that is why, or you can say chlorophyll. Okay. You can simply write they contain both chlorophyll and carotenoids. So although they contain both chlorophyll and carotenoid, but the predominant green color is of the chlorophyll. That is why these plastids are green in color. Okay, they are found in leaf mesophils. Very common in leaf mesophyll cells. These green plastids are found. Now chromoplast, they contain carotenoids, but they do not contain chlorophyll. That is why green color does not come. They contain carotenoids. Now, they can be either orange or they can be red, they can be yellow. Nowadays, if you see capsicum in the market, earlier only green capsicum used to come. But now if you go to market, you see different bright color capsicum is coming, brightly red, brightly yellow. Okay, so this is if the plastid has only carotenoid but no chlorophyll, then it is a color plastid. As you can see, the carrot, the orange color of the carrot you see, or when the chilies or tomatoes, when they ripen, you see they convert into red color. It means one more thing is also there, that one type of plastid get converted into another type of plastid, because as the chilies are maturing, the chloroplast is being converted into the chromoplast. Fine. Okay. Next, leucoplast are colorless plastids. No pigments. Due to absence of any pigment, these are colorless plastids. Why isn't nucleus included in endomembrane system? Has roles like linked with ER and genome required for protein synthesis? No, actually you see endomembrane system where there is a continuity. Okay, nucleus is not in that continuity. I told you, no, that what will happen? Protein produced on ER, elaborated in Golgi complex, form a lysosome formed, and lysosome will go to the target to digest. This continuity end. Nucleus does not come in between. That is why nucleus is not a part of the endomembrane system. Fine. Okay. Haji, no pigments. That is why they are colorless. But mainly function is storage. Now, on this basis, leucoplast is divided into three types. If it is stores starch or carbohydrate, then amyloplast, for example, potato tuber. Alluron, you know, protease, if it stores protein, then it is alluroplast. You have seen in maize seed alluron layer. And elioplast stores lipid. For example, fatty seeds, mustard. Okay, elioplast. Okay. Nucleus is a cell organelle. Nucleus is the largest cell organelle. Nucleus is the largest cell organelle. Yes, nucleus is a cell organelle. Okay, fine. Fine, children. So this is all regarding the. Now let us confine ourselves to the chloroplast. Chloroplast is a double membrane bound structure, outer membrane, inner membrane, and interior of the chloroplast has two parts. The interior of the chloroplast has two parts, a fluid part which is called as the stroma, and a membranous part which is called as thylakoids. Now these thylakoids are again of two types. If the thylakoids are stacked one over the other, they are called as grana. And if they are not stacked in between the grana, these thylakoids are called as the stroma lamella or stroma thylakoids. The grana is the site, the stroma is the site of the dark reaction of photosynthesis, and thylakoids, that is the grana and the stroma lamella, are the site of light reaction of photosynthesis. Just like the mitochondria, chloroplast also has 70S ribosome and circular double-stranded DNA. So that is why chloroplast can also synthesize some of its proteins itself. That is why chloroplast is also a semi-autonomous organelle. Fine? Okay. Now coming to ribosome. Eukaryotic ribosomes are larger in size. They have high sedimentation coefficient. Density is measured in S unit, which is called as Swedberg unit or sedimentation coefficient. So in eukaryotic ribosomes, they are called as the 80S ribosomes. The smaller subunit is called as 40S and the larger subunit is called as 60S. And as I said, this 80S ribosome and the Swedberg unit stands for the sedimentation coefficient. It is the indirect measure of density and size both. Remember, it is the measure of both density and size of the ribosome. And ribosomes are the site for the protein synthesis. It was discovered by George Palade, and these are also called as the protein factory of the cell. Now, there are certain proteinaceous network. This proteinaceous network present in the cell is called as the cytoskeleton. Cytoskeleton is of three types. Okay. Microtubules. And you know these microtubules are important for the formation of spindle fibers during cell division. Microfilaments and intermediate filaments. Filaments. Three types. And their main function, this question was asked last year. Function was 2023's question was asked. Their function is mechanical support, motility. The cytoplasmic streaming that you see in the cell, it is due to, okay, maintenance of shape of the cell. These are the main functions. Okay, fine. Sir, please give clarity about fat-soluble plastids. Fat-soluble plastids? No. Sorry, I'm sorry. Plastids are not fat-soluble. Carotenoids are fat-soluble. Please read again. They are saying they contain fat-soluble pigments. Carotenoids and chlorophyll does not dissolve in water. So chlorophyll and carotenoid you can dissolve in the, okay, fat. That is why they are called as fat-soluble. Okay, understood? And phycobilins you can dissolve in hot water. Understood? Jagan. Yes. So children, this is cytoskeleton. Next we come to cilia and flagella. You see, both cilia and flagella have same basic structure. Thank you. Both cilia and flagella have same basic structure. The difference is size. Cilia are smaller while flagella are longer in size. Cilia number per cell is very high. Flagella number per cell is very, maybe 1 or 2 or 4 more than that, not more than that. If you see paramecium, it has cilia all over the body surface. But if you see euglena or if you see chlamydomonas, only 2 flagella. Dinoflagella, it's only 2 flagella. Or maybe it up to 4. Green algae have up to 4, but longer in size. Otherwise, difference. Main function of both cilia and flagella is locomotion. But cilia, as you have seen in case of the paramecium, it also helps in trapping the food. Okay, in trapping the food also, but the main function is locomotion, but cilia also help in food trapping. Now coming to the structure. Both cilia and flagella have, I said, same basic structure. A basal body which is embedded in the cell membrane. Basal body which is embedded in the cell membrane and a filament. Now, the NCERT has drawn the structure. Hydra, you ask your zoology teacher. I don't know about Hydra. Okay, fine. So, filament, it has central axoneme, which is made up of protein and plasma membrane. So, the transverse section of this filament is given in your NCERT. Let us try to see this. See here. So this is the plasma membrane, this entire is the axoneme. Now axoneme has two central microtubules or central microtubules which are singlet and 9 doublet, 1, 2, 3, 4, 9 peripheral doublet microtubules, fine. So that is why it is called as 9 plus 2 arrangement, 9 doublet peripheral and 2 singlet. The two central singlet are connected to each other by a bridge and the central filaments are surrounded by a central sheath, you can see. The peripheral doublet filaments are also connected to each other to interdoublet bridge. You can see here interdoublet bridge. Okay. Now, and the peripheral filaments are connected to central sheath through spokes, which are called as radial spokes. So, number of radial spokes will be equal to the number of peripheral filament. Okay. So, plasma membrane. Under the plasma membrane, there are 9 doublet peripheral filaments, okay, which are connected through interdoublet bridge. Then there is a central sheath, and central sheaths around the 2 central singlet filament. Is this structure clear to everyone, please? And it is common for both cilia and flagella. Now, coming to centrosome and centriole. Centrosome and centriole I will show you. A centrosome has 2 centrioles. Acha, once again, okay. Haji, see. This is the transverse section of the filament. The outermost covering is the plasma membrane, which surround the central axoneme. The central axoneme has 9 peripheral doublet microtubules, and they are connected to each other through interdoublet bridge. Okay, and 2 central singlet microtubules, which are not double. Okay. Now the peripheral microtubules are separated from central microtubules by a sheath, which is called as the central sheath. That is all. Fine. Both chloroplast and mitochondria have 70S ribosomes. The endoplasmic reticulum has 80S ribosomes, but within the chloroplast and mitochondria, both there are 70S ribosomes. That is why, although we say eukaryotic ribosome 80S, but eukaryotes also contain 70S ribosome, but only within the organelles like chloroplast and mitochondria. Okay, fine. Haji, next. Centrosome. You see, centrosome contains 2 such tubular structures surrounded by a pericentriolar matrix, and these are centrioles. Centrosome. Okay, so this is called a centrosome. These are found only in the animal cells, and they help in the formation of the spindle apparatus during the cell division in animal cells. If you see each centriole, it is like this. There are no central microtubules. There are 9 radial spokes. Each radial spoke has triplet of microtubules.
Okay, and these are connected like this. So this gives a cartwheel appearance like a cartwheel. That is why these are called as cartwheel organelles. Fine. And I've already told you that they help in the formation of a spindle apparatus during cell division. And one more thing, the centrioles, they form the basal body of cilia and flagella. Okay. So they form the basal body of cilia and flagella. So this is all regarding the centrioles. Next, we come to nucleus. Nucleus, I've already told you that nucleus has a nuclear envelope which surrounds the nucleoplasm. The nuclear envelope is a double membrane bound structure. On the outer surface, it has 80 S ribosome. Remember, on the outer surface of the outer membrane, there are 80 S ribosomes, and outer membrane is also in contact with the endoplasmic reticulum. Okay, outer membrane is also in contact with the endoplasmic reticulum. Fine. And in between the two membranes, there are certain pores which are called as nuclear pores, and through these nuclear pores, there is both way movement of RNA and protein. Okay, both way RNA and protein can move both way between the nucleoplasm and the cytoplasm through the nuclear pore. Now, the nucleoplasm contains mainly two things, nucleolus and the chromatin. The nucleolus are non-membrane bound structures. Remember, nucleolus is not organelle. Organelle is a nucleus. Within that, non-membrane bound structure is present, which is called as nucleolus. And since it has no membrane, it is in continuation with the nucleoplasm. Nucleolus is the site of synthesis of the ribosomal RNA. That is why a cell which is actively involved in protein synthesis, they have large number of and large size of the nucleolus. Fine. So nucleolus, basically protein synthesizing cells will have large number and large size of nucleus. Now coming to chromatin, okay. This chromatin is the association between DNA and histone protein and also some amount of RNA. So let us see. The interphase nucleus has a loose and indistinct network of nucleoprotein fibers called as chromatin. And when this chromatin condenses, it forms chromosome. So chromosome is nothing but the condensed form of chromatin. Now, what is the chemical composition? Let us try to see. Okay, this chromatin consists of DNA, histones, some non-histone proteins, and also RNA. Okay, a single human cell has 46 chromosomes, and in 46 chromosome, there are around 2.2 meter of total DNA present in this 46 chromosomes. Now coming to the chromosome structure, as I said, chromosome is nothing but the condensed form of chromatin. For study, we see metaphasic chromosome because metaphase is the phase in which chromosomes show maximum condensation. So that is why metaphase is the best phase to study the chromosome structure. A metaphasic chromosome consists of two thread-like structures called as chromatids. Chromatids. Okay. At one place, the two chromatids are connected to each other. This is called as centromere or primary constriction. Okay. And on the centromeres, there are proteinaceous structures which are called as kinetochore for attachment of spindle fibers during cell division. Attachment of spindle fibers during cell division. Okay. Apart from this, certain chromosomes have one additional constriction which is called as the secondary constriction. You can see this is called as the secondary constriction. This labeling is slightly wrong, children. This line should have been here. And the portion beyond the secondary constriction is called as satellite. And this satellite is very poor in DNA. That is why when we stain the chromosomes, satellite does not get stained. Okay. Secondary constriction are the site for the formation of nucleolus.
But remember, secondary constriction is not present in all chromosomes. Very few chromosomes have secondary constriction, while primary constriction or centromere is present in all chromosomes. Now, children, the position of the centromere determines the shape of the chromosome. And this shape can be observed at the anaphase. Okay. If the centromere is totally exactly in the center, then you will see V shape at the anaphase, and such chromosomes are called as metacentric. Metacentric. If the centromere is slightly away from the center, then you will find one arm slightly shorter than the other arms. Then this is called as submet. This will show L shape, and this is called as submetacentric. Submetacentric, fine. Okay. So both these are L shape and submetacentric. If one arm is very short and one very long, then it takes J shape, this one, J shape, and this is called as acrocentric. And if the centromere is terminal, then it is called as telocentric. Okay. Which is not drawn here. This is the telocentric chromosome. This shape, I shape, and this is called telocentric. So depending upon the position of the centromere at anaphase, we can shape 4 types of shapes of the chromosome: metacentric, submetacentric, acrocentric, and telocentric. Fine. So this is all regarding the chromosomes. Secondary constriction I've already told you. Last thing is microbodies. There are certain single membrane bound structures in the cell. Regarding C-value paradox, who asked you to study C-value paradox, Jagan? 2C, 4C, that we will do in cell division, no? Don't worry. That we will do in cell division, fine. Okay. So, children, microbodies, there are single membrane bound organelles, certain which are collectively called as microbodies. These are of 3 types. First is peroxisome. These are found in both plants and animals. They contain mainly two enzymes. One is the peroxidase and one is catalase. And in animal cells in our body, they help in detoxification of drugs and toxicants, pollutants, and even alcohol. They help in all this detoxification. And peroxisome, in case of a plant cell, they are involved in photorespiration, if you remember it. Fine. Then second is glyoxysome. They contain the enzymes of glycolate cycle. Okay, they contain enzymes of glycolate cycle by which they convert fat into carbohydrate. And third is spherosomes. They store lipid. Though these are found in fats, fatty seeds. Where food is stored as lipid, they are found in fatty seeds. These are spherosomes. So these are collectively called as microbodies. So we have completed the chapter Cell, the Unit of Life. And next we will be doing is the biomolecules. Okay. But before that, a good water break. Huh. Microbodies are single membrane structures or not? Yes, they are single membrane. Achha, you get confused due to the NCERT language. They have written many membrane bound. So people think that they have many membranes. They are not saying, they are saying many means many types of membrane bound organelles. Fine, understood? Pusala. Pusala, is it clear? They are saying there are many, comma, membrane bounds. Okay. हाँ जी, biomolecules. Sir, centriole में 9+0 arrangement and basal body of cilia and flagella. Yes, very good question she has asked. Just a minute, please let me finish the water and coming. Very good question. If you are asked, children, what is the arrangement of microtubules in the filament of cilia and flagella, your answer will be 9+2. But if you are asked what is the arrangement of microtubules in the basal body of cilia and flagella, since basal body of cilia and flagella are made up of centrioles, you will say 9+0 arrangement. I think Sanapala Reshma. Reshma, have you understood what I said? Okay. So children, let us start with the biomolecules. Carbon containing organic compounds found in living beings. Biomolecules are of two types. Micromolecules means molecular weight less than 1000 Dalton. वैसे generally we say that 18 to 800. 18 to 800 we say, if you want to go to strictly 18 to 800, but we can say less than 1000, that is biomicromolecules. Example, amino acid, then lipids, nucleotides, and monosaccharides. And biomacromolecules. Biomacromolecules. Molecular weight much more than 1000 Dalton. Much more than 1000 Daltons. Okay. Biomacromolecules. Now polymers of amino acid, that is proteins. Next, polymers of nucleotide, that is nucleic acids. And polymers of monosaccharide, that is polysaccharides. But children, lipids do not form polymers. Very, very important question. It is very commonly asked. Lipids do not form polymers. That is why lipids can never be macromolecules. They do not form polymers. Fine? Okay. So, but children, when we try to filter them, okay, Suppose we are using a filter. We chloroacetic acid. Suppose we try to dissolve it into, and then we will get acid insoluble and acid soluble. Acid insoluble, of course, there will be proteins, then nucleic acids, polysaccharides, and acid soluble will be amino acids, monosaccharides, nucleotides. But interestingly, lipids come here. Lipids are not true biomacromolecules, but when we filter them, they are separated along with biomacromolecules. The reason is children, that lipids, they form the cell membrane. And when the cell is disrupted, these membranes form large vesicles. Okay, and that is why they do not get filtered through the cheesecloth, and they are retained on the surface. So although lipids are not true biomacromolecules, but during filtration, they are separated along with biomacromolecules. Reason is they form the cell membrane lipid bilayer, and when the cell membrane is disrupted, the fragments of lipid form vesicles, and they do not cross the cheesecloth, and they are filtered along with the biomacromolecules. Is it clear? So this is one exception. Fine. Now let us come to each one by one. We are starting with biomicromolecules. These tables, children, please remember all these tables. Okay. No one knows where they will ask from. First, amino acids are organic compounds containing an amino group and an acidic group as a substitute on the same carbon. And this carbon is called as alpha carbon. So if both amino and the acid group are present on same carbon, that are called alpha carbon, and such amino acids are called as alpha amino acids. Please remember this. Okay, fine. That is the alpha carbon. Hence, they are called as alpha amino acid. They are substituted methane. We say that amino acids are substituted methane. Let us see why. See here, this is methane, CH4. Now, suppose one hydrogen is replaced by a amino group, another hydrogen is replaced by acid group, another hydrogen is replaced by R functional group, and only one remains. So see, this H replaced by amino group, this H replaced by R group, and this H substituted by acid group. That is why we say that amino acids are substituted methanes. Fine. Okay. And you can see they have a one amino group, one acid group or carboxylic group, and one R functional group. So the main difference among amino acids is in the type of R group. Okay. So different amino acids differ basically due to their R groups. Let us try to see this. See here. These are 3 amino acids. Here, the R group is very simple, hydrogen only. That is why glycine is called as the simplest amino acid. The most simple amino acid is glycine because the R group is H. Alanine, see, all things are same. The R group is methyl. And here, the R group is CH2OH. So see, so serine, glycine, alanine, and serine. So main difference is in the R group, and the most simplest amino acid is the glycine because R group is a hydrogen. Fine. Next. The chemical and physical properties are essential by carbon R group family. Based on number of amino and carboxylic group, there can be acidic amino acids. If the carboxylic group is more, naturally it will be acidic. So acidic amino acids are glutamic acid. Okay. Basic amino acids, where the amino group is more, lysine, and neutral, where equal, both amino and carboxylic group are equal, that is called as the neutral amino acid. So valine is a neutral amino acid. Lysine, if you remember, you have already said that histones are basic proteins because they contain basic amino acid like lysine and arginine. So that you have already studied. Fine. Okay. So on this basis, amino acids can be either acidic or basic, or they can be neutral. Next is that in some amino acids, instead of the R group, there is a aromatic ring. These are called as aromatic amino acid, like tyrosine, tryptophan. There are aromatic amino acid because in place of R group, there is a aromatic ring. So please remember the name of the aromatic amino acids also. That is also important. Fine. Now, in solutions of different pH, the amino group can also be ionized and carboxylic group can also be ionized, and both can even be ionized. So here if you see first one, there is ionization of the amino group while carboxylic group is normal. In second case, there is ionization of the carboxylic group or acidic group while amino group is normal. And in the central case, you see both are ionized. So in an amino acid, if both amino and carboxylic group are ionized, such form is called as Zwitter ionic form. So Zwitter ionic form amino acid is that amino acid in which both amino and carboxylic group are ionized. Fine. So children, this is all regarding the amino acids. Next we come to lipids. Okay, lipids do not form polymers and lipids do not generally dissolve in water. You see, if you try to dissolve oil in water, it will not dissolve. Okay, so lipids generally they are soluble in organic solvents, not in water. Fine. Okay, so lipids, what come under lipids? Let us try to see. Lipid is a very long range. Fatty acids. They all come under lipids. Fine. Now fatty acid, coming to fatty acid, okay, they have R and COOH. Okay, this R has up to 19 carbon. Okay, so a fatty acid can have maximum 20 carbon, 19 in the R group and 1 in the COH. So including the COH, it can have maximum 20 carbons. Okay, this R can be an ethyl group, R can be a methyl group, or it can be a long chain of CH2. So R can be ethyl group, R can be methyl group, or long chain of CH2. Okay, this is called as the fatty acid. And I said fatty acid, the R group can have maximum 19 carbon. So including carbon of the COH, 20 carbon maximum in fatty acids. For example, the palmitic acid has 16 carbon. Okay, stearic acid has 18 carbon and arachidonic acid has 20 carbon. So all this you have to remember. These questions are very frequently asked. Now, if a fatty acid has no double bonds, then it is called a saturated fatty acid. And if even one double bond is present, then it is called as the unsaturated fatty acid. Okay, that unsaturated fatty acid can be MUFA, that is mono unsaturated fatty acid, or it can be PUFA, polyunsaturated fatty acid. They are different types. I'm not going into those details. Fine. This unsaturated fatty acid, we say it is good for health. It is good for health. Okay, fine. So these are all regarding the fatty acids. These numbers are given, these questions, from this part, very commonly questions are asked. Saturated fatty acid, no carbon-carbon double bond. Unsaturated fatty acid, carbon-carbon double bond present. Okay, fine. Now, as I said, the esters of fatty acid, glycerol is also, I have written here, just a minute. Glycerol is also a fat. Glycerol are trihydric alcohols. Let us see. So see, again we are referring to this. See here. This is a fatty acid. You can see this is the R group and this is the COH. Okay. This is a glycerol. Okay. Trihydroxypropane. This is glycerol. And this are glyceride. Glycerol, if you see here clearly, this is the glycerol. This is one fatty acid. This is second fatty acid. This is third fatty acid. And this bond is the ester bond. So this is a triglyceride because one glycerol, three fatty acid. If there is one glycerol, one fatty acid, it will be called as the monoglyceride. One glycerol, two fatty acid, it will be called as diglyceride. And one glycerol, three fatty acid, it will be called as triglyceride. Maximum it can be triglyceride, fine. And this bond between the fatty acid and glycerol is called as the ester bond. These are simple fats. Then fats can be other complex fats also. For example, if you see this one, apart from this is one glycerol, two fatty acid, but apart from this, it also has a phosphate group and a nitrogen containing compound choline. Okay, so these are the complex fats. Apart from the fatty acid and glycerol, they contain other compounds also, and this is called as phospholipid lecithin, which is the constituent of the cell membrane. So the double layer that we saw in the cell membrane, that is of the phospholipid lecithin. It is made up of, fine. It is a nitrogen containing and phosphorus containing lipid, phospholipid, fine. Okay. And if sugar is attached to any lipid, then it will be called as glycolipid. Fine. So this is all regarding children lipids. One more thing, that fat and oil, this definition is also done on the basis of what is the consistency in winter. For example, the gingerly oil, okay. The gingerly oil has low melting point. So in winter also it is a liquid that is called as the oil. But like the butter, in winter it is solid, then it is called as fat. So this is also one of the criteria for classification. Let us see here. See, these are also called fat and oil based on the melting point. Oils have lower melting point. Gingerly oil hence remain as oil in winter. While, as I said, butter or coconut oil, you see in winter they become solid. Okay. Some lipids have phosphorus and organic compound in them. These are phospholipids. They are found in cell membrane. Lecithin is one of them. Some tissue, especially, this is important. Neural tissue have lipids with more complex structures. Okay. Then nucleotides. Nucleotides have three things. One, heterocyclic compound, which is a nitrogenous base. These are of two types, purines, adenine and guanine. Pyrimidine pyrimidines, thymine, cytosine, uracil. Adenine and guanine are present in both DNA and RNA. Okay, cytosine is also present in both DNA and RNA, but this only in DNA and this only in RNA. Fine. So this is heterocyclic compound. Second is monosaccharide. This is ribose sugar, pentose sugar. Ribose sugar in RNA and deoxyribose in DNA. Fine, deoxyribose in DNA. And third component is phosphoric acid. Phosphoric acid. So these are the three components of a nucleotide, and it is something like this. This is the pentose sugar. This is the nitrogenous base, and this is the phosphate group. This bond is called as N-glycosidic bond, and this bond is called as phosphoester bond. Okay, phosphoester bond. So these are, children, the different types of biomicromolecules that we have studied. Coming to biomacromolecules. Let us do the monosaccharides also here, biomicromolecules. This monosaccharide, if you see here, this is a 6-carbon monosaccharide, which is glucose, and this is a 5-carbon monosaccharide, which is the ribose. Okay, so ribose sugar, this is present in the DNA and RNA, ribose and DNA, this oxygen will be absent. This is called as the deoxyribose sugar. Fine. So this is glucose and monosaccharides. Next we come to one more thing. I told you that the fats also include, lipids also include sterols. Sterols, the cholesterol is one of the sterols. They are not true fats, okay, because they do not have fatty acid, but they show features similar to the fats. That is why they are included in the lipids. And cholesterol, I told you, it is present in our cell membrane and it helps in maintaining the fluidity. Apart from that, cholesterol is also very important, okay, for the synthesis of vitamin D in our body. So we generally think that cholesterol is very bad. It increases the blood pressure. Okay, excess of anything is bad. Cholesterol is important for synthesis of vitamin. We say sunlight. Sunlight is only provides the stimulus. Okay, the vitamin D is formed from the cholesterol. Fine. Okay. Now, we have talked about the nitrogenous base. Purines. Purines have 2 rings. Okay. And pyrimidines have single ring. Okay. So this is adenine because at this position there is amino. This is uracil. Okay. This is uracil. This is the pentose sugar. Now, if nitrogenous base plus pentose sugar, then it is called as a nucleoside. And nitrogenous base plus pentose sugar plus phosphate group, then it is called as nucleotide. So nitrogenous base plus sugar, nucleoside. Nucleoside plus phosphate group is called as nucleotide. Fine. Okay. So this chart we have completed. Remember, children, that this table is very, very important. Very important table. Please remember it. Every year there is a question on this. Fine. So there are certain chemicals which are produced by plants and not produced by animals. Plant, bacteria, microbes, they produce, fungi, they produce certain chemicals, what is exact physiological role in the body of the organism that is producing, we at present do not know. So these chemicals are called as secondary metabolites, but they are of great economic importance to us. Okay, they are even of ecological importance. And these secondary metabolites, the list is given here. See? So please remember this list of secondary metabolites, very, very, very important. Fine. Next, biomacromolecules, we are coming. Biomacromolecules, okay, as I said, that 18 to 800, this is biomicromolecules, okay. not exceeding 800 biomicromolecules, and more than 1000, even much more than one, then it is the biomacromolecules. Now, this table is also important. Please remember water, carbohydrate, and all that. Okay. Now coming to proteins. Proteins are polymers of amino acids. They are connected through the peptide bonds. Polymers of amino acid connected to peptide bonds. Now, amino acids are of two types. The amino acids which are required by our body, but they are not synthesized by our body, they are called as the essential amino acids that we get through our dietary supplement. There are many, 8, 9, something like that. Okay, fine. They are essential amino acids. Other we are able to synthesize. Now, collagen is the most abundant animal protein which forms the intercellular ground substance. But overall, the most abundant protein on earth or in the entire biosphere is the enzyme ribosome, which is found in plants, and you know it is the first enzyme of the Calvin cycle. Fine. Okay. Now, these are given. This GLUT4 has been asked twice. Okay. So this protein, it is a membrane protein that helps in the transportation of the glucose into the cell. Okay. It provides facilitated diffusion to the glucose in the cell, GLUT4. Fine. Okay. Now, essential, non-essential we have done. Collagen is the most abundant protein in animal world and ribosome is the none. Now, polysaccharides. Polysaccharides are the polymers of monosaccharides connected through glycosidic bond. Okay. Polymers of polysaccharide connected to glycosidic bond. Now, polysaccharides can be homopolysaccharide or heteropolysaccharide. If a polysaccharide is made up of only one type of sugar, it is called as the homopolysaccharide. For example, cellulose. Cellulose is a homopolymer of beta glucose. Okay. You know that if the first carbon OH is above the plane of the ring, it is called as the beta glucose, and below the plane of the ring, it is called as alpha glucose. So it is a homopolymer of beta glucose. Cellulose does not show complex helical structure, so it cannot retain iodine. It does not give a positive iodine test. Cellulose is a structural polysaccharide. Why? Because it forms the cell wall of the plant cells. Okay. And it also forms the cotton fibers, paper, all these are cellulose fibers. Fine. Next, starch. Starch is a homopolymer of the alpha glucose. Okay, it is a homopolymer of alpha glucose. It is a storage polysaccharide because plants store food in the form of the cellulose. Starch. Similarly, glycogen is also a homopolymer of the alpha glucose. The very basic, some small differences between the starch and the glycogen. Both are homopolymer of alpha glucose. Both are storage polysaccharides. Plants store in the form of a starch, animals store in the form of glycogen. The difference is in their branching pattern. Okay, branching pattern. Okay. Now, starch shows complex helical structure, so it retains iodine and gives blue color. So that is why it gives a positive starch iodine test by giving blue color. Right. So that is why we say that the starch and glycogen are variants of glucose. Variants of cellulose. Why variant? Because cellulose is also a homopolymer, but it is a homopolymer of beta glucose. And starch and glycogen are homopolymer, but they are the homopolymers of the alpha glucose. Now, there is a fourth one, which is inulin. Inulin is also a storage polysaccharide because plants like the Dahlia store food in the form of inulin. But inulin is a homopolymer of fructose. While cellulose, glycogen, and starch are homopolymers of glucose, inulin is a homopolymer of fructose. Okay, and it is also a storage polysaccharide because plants like Dahlia root store food in the form of inulin. These are simple polysaccharides. Now we come to complex polysaccharide. Complex polysaccharide have some other things attached to it. For example, N-acetylglucosamine. Now either sugar, okay, N group is also attached, nitrogenous group is also attached. That is why it is called. For example, chitin. Chitin is a homopolymer of NAG. It is a homopolymer of NAG. Chitin is also a structural polysaccharide because chitin forms the cell wall of fungi, and chitin is also found in the exoskeleton of insects. So, okay. So these are the different types of polysaccharides which we have discussed. I'm just giving a detail again. Okay. Cellulose, homopolymer of beta glucose, structural polysaccharide, forms cell wall of the plants. Starch and glycogen, they are homopolymers of alpha glucose. They are storage polysaccharide because Plants store food in the form of a starch, animals store food in the form of glycogen. Then inulin, it is a homopolymer of the homopolymer of fructose, and it is also a storage polysaccharide because, as I said, some plants store in their root food as this. Fine. Now, the complex polysaccharide or chitin is a very good example of, because it is a polymer of the NAG. Okay. Even the cell wall of the bacteria, NAG, NAM, these are all examples of the complex polysaccharides. Fine. Let us see this last point and then we can move forward. You see, this is the branching pattern. Both glycogen and starch show branching, but the frequency of branching is different between diagrammatic representation of a portion of glycogen is given here. Fine. Starch will also show, but branching pattern is slightly different. Remember this bond, children. This bond is called as alpha 1,4 glycosidic bond. So in the straight chain, the bond, this is called as alpha 1,4 glycosidic bond. But here you see, this bond is called as alpha 1,6 glycosidic bond, because bonding is between the first carbon of this and sixth carbon of this. That is why first carbon of this and sixth. That is why alpha 1,6 glycosidic bond. And here you see between 1 of this and 4 of this. That is why this is called as alpha 1,4. So remember, in a polysaccharide like starch or glycogen, the bond between the straight chain is alpha 1,4 glycosidic bond, and the bond at the branches is alpha 1,6 glycosidic bond. So this is alpha 1,4 glycosidic bond, and this is alpha 1,6 glycosidic bond. Fine. Okay. See here. There are more complex polysaccharides in nature. They have as building blocks amino sugars and chemically modified sugars. Glucosamine, acetyl glucosamine. Exoskeleton of arthropods, for example, have a complex polysaccharide chitin. These complex polysaccharides are mostly homopolymers. Fine. Now, structure of nucleic acids I am not discussing because already it will be discussed in your genetics portion, so we can save some time for other things. Fine. See here, structure of protein, a long chain of amino acid. This is called as primary structure. On left hand side you will have an N terminal, on right side you will have C terminal, because here a free amino will be present and here a free carboxylic will be present. Fine. Now this primary structure, either it can spiral like this, then it is called as alpha helix, or it can be folded like this, then it is called as beta plate. So both alpha helix and beta plate are examples of secondary structure of protein. Okay, and when it is folded upon itself, this chain of polymers, polypeptide chain is folded upon itself like a hollow wool of ball, then it is called as the tertiary structure. And during tertiary structure, other types of bonds are also found, like hydrogen bond, disulfide bond, other kind. Hydrophobic bond may also be found. And when more than one such hollow balls, they connect to each other, then it is called as the quaternary structure. So single hollow ball like structure is tertiary. But when many such hollow ball like structures, suppose 1, 2, 3, linear arrangement. This is a quaternary structure. Many shapes they can arrange like this. These are all quaternary structures. And a very good example of quaternary structure is the hemoglobin. Okay. Hemoglobin, it has 2 alpha chains and 2 beta chains. Okay. So it is a tetramer, but 2 alpha chains and 2 beta chains. See here. Quaternary structure. 4 subunits, 2 of these are identical to each other. Hence, the 2 subunits of alpha type and 2 subunits of beta type together constitute the human. Adult human hemoglobin has 2 alpha and 2 beta. Fine. Nature of bond linking, simple, I am telling you. Between 2 amino acids, the bond is called as the peptide bond. Okay. Between 2 nucleotides in a DNA or RNA, the bond is called as the phosphodiester bond. Between the fatty acid and glycerol in a fat, the bond is called as the ester bond. Sir, how many reasons are there for denaturation of protein? Jagan, I am not able to understand your question. What do you mean by this? I think you are making some very advanced studies. Generally, if you increase the temperature above 40 degrees, most of the proteins are denaturated because their folding pattern changes. Okay, but there can be many reasons. Okay, fine. Water break. That we don't teach. That you study in chemistry. That coagulation, isoelectric point, all these you study in organic chemistry. That is beyond my scope of teaching. Okay, ji. Fine. So in a nucleic acid, the bond between adjacent nucleotides is called as the phosphodiester bond. Okay, phosphodiester bond. So these are the bond linkings. This metabolism portion we have to leave. Okay. Now coming to enzymes. Enzymes are the catalyst of the biological system. Okay, enzymes, they increase the rate of reaction. Here, important point I will discuss is this remember how enzymes increase the rate of biochemical reaction. We have to understand. Suppose enzyme has places called as active sites. The substrate binds to the active site of enzyme to form an enzyme substrate complex. Okay, then the existing bonds are broken and new bonds are formed. As a result, the enzyme substrate complex converts into enzyme product complex, and then the product and enzyme get separated. And during this process, ultimately the enzyme itself finally does not change. So enzyme can be reused. Fine. Okay, something like this. See here. See. Substrate will bind to the active site of the enzyme to form enzyme substrate complex. Then the existing bonds will be broken and new bonds will be formed to form the enzyme product complex. And finally, enzyme and product will be separated. This enzyme can be reused. Fine. Now, children, what happens? Remember, no enzyme can initiate a chemical reaction. Enzyme can only alter the rate of the reaction. Fine. How? Let us see. You see, whether exothermic reaction or endothermic reaction, always if the exothermic reaction, okay, then the energy level of product will be less than that of the substrate because energy has been released. If it is endothermic reaction, then energy level of the product will be something like this, more than the substrate because you have given energy. But you have to understand whether it is exothermic reaction or endothermic reaction, substrate never directly get converted into product. Between substrate and product, there are one or more than one intermediate transition states, okay, which are called as the transition state. And to reach this transition state, very large amount of energy is required, which is called as the activation energy. Activation energy. And this activation energy is required for both exothermic as well as endothermic reaction. Due to this very heavy requirement of energy for the conversion of substrate into the transition state, very less substrate get converted into transition state. That is why the rate of product formation is very, very low. Okay, that is why rate of reaction is low. But when enzyme is present, enzyme lowers this activation energy. So now very less energy is required for the substrate to reach the transition state. So due to the lowering of the activation energy requirement, many more substrate molecules get converted into product. So enzymes increase the rate of a biochemical reaction by lowering the activation energy. Fine. This is what has been shown in this graph. See, this is the energy level of substrate. Now, you see red one. To reach transition state, this much energy was required. So the difference of energy level between the substrate and transition is called as the activation energy. So in the absence of enzyme, this much energy was required to reach the transition state. And due to this high demand of energy, very few substrate molecule got converted into product. But when enzyme was present, in the presence of enzyme to reach the same transition state, now very less energy is required. You see, energy requirement only this much. So due to this low requirement of energy to reach the transition state, many more substrate molecules got converted into product. As a result, the rate of biochemical reaction increased. Is it clear to you, children? So enzymes increase the rate of reaction by reducing the requirement of the activation energy to reach the transition state. Once it reaches the transition state, then it will be converted into the product. Is it clear? Now, see these graphs. Effect of temperature or pH. Every enzyme functions as a very narrow range of pH. So if this beyond this range also temperature rate will decrease, and before the also rate will become and less. Same will be temperature. Every enzyme shows its best activity in a narrow range of temperature. So low temperature enzyme will remain inactive, high temperature enzyme will denaturate. Okay, so that is why both will show bell-shaped curve. But coming to substrate concentration, S means substrate concentration. As you will increase substrate concentration, more and more active sites will get occupied. So the rate of reaction will increase, keep on increasing. But a time will come when all active sites are occupied. No active site is free to bind to the substrate. Now, if you increase further substrate concentration, there is no increase in the rate of reaction. Why? Because the reaction has reached saturation. Okay. At this time, we say that the enzyme is working at its maximum velocity called as Vmax. Okay, half of Vmax, this is called as Vmax by 2. Now, that substrate concentration at which enzyme had reached Vmax is called as Michaelis-Menten constant or Km. So Km is that substrate concentration at which enzyme reaches half of its maximum velocity, that is Vmax by 2. Why we calculate Vmax by 2? Vmax by 2 gives us the reference of the affinity of the enzyme for the substrate. More the affinity of the enzyme for the substrate, very easily it will reach Vmax by 2. So means by using lesser the Km, more is the affinity of the enzyme for the substrate. Because by using lesser substrate, enzyme is reaching very early Vmax by 2. Means enzyme is getting more active by using less substrate. So Km is calculated to know the affinity of the enzyme for the substrate. Now coming to substrate inhibitors. Inhibitors are chemicals that inhibit the activity of the enzyme. Here we are talking about only one inhibitor, that is the competitive inhibitor. Competitive inhibitor is a chemical that competes with the substrate to bind to the active site of enzyme. But problem is, if inhibitor binds to the active site of the enzyme, it will occupy this active site, but it will not form product. So as a result, the free active sites available for the substrate to bind to the enzyme will decrease. As a result, the rate of reaction decreases. Okay. So that is why it is called as competitive inhibition, because the substrate is chemically and structurally similar to the substrate. That is why it is competing with the substrate. For example, see here. Suppose this is an enzyme. This is the active site. If substrate is binding to it, then there is formation of product. But if inhibitor is binding to the active site of enzyme, no product formed. So if more and more inhibitor will bind to the active site, means the free active site available for substrate binding will decrease. As a result, the enzyme activity will decrease, and this is called as competitive inhibition. Such competitive inhibition are used to check bacterial pathogens. Okay, Because what is done, the bacteria has to produce its chemical, like folic acid bacteria will produce by using an enzyme. We give sulfa compounds which are competitive inhibitor of the enzyme that produces folic acid in bacteria. So bacteria will not be able to produce its folic acid and it will die. So this is how we can control the bacterial pathogens. A very good example of the competitive inhibition is when you study Krebs cycle. Enzyme succinate dehydrogenase, okay, which converts the succinate into fumarate. Its competitive inhibitor is malonate. So if you give malonate, the rate of Krebs cycle will decrease because it will decrease the activity of the succinate dehydrogenase enzyme. Very, very common question. Remember that malonate is competitive inhibitor of the enzyme succinate dehydrogenase that converts succinate into fumarate. Fine. So this is all regarding the factors affecting enzyme activity. Last thing we are coming is cofactors, because cofactors I have found students having problem. So remember, most of the enzyme to become active, they require a non-protein. We say almost all enzymes are proteinous. Exception are few cases where RNA functions as enzyme, for example, peptidyl transferase. So when RNA is enzyme, it is called ribozyme. But mostly enzymes are proteins. But most of these enzymes, they require one non-protein part also to become active. This non-protein part is called as the cofactor. So remember, apoenzyme plus cofactor. This is the protein part or protein moiety. This is the non-protein part. Together they form the holoenzyme. Active enzyme. Okay. Remember, this equation is also asked. Apoenzyme plus cofactor is equal to holoenzyme. Okay. So this is the non-protein part. This cofactor is of three types. If it is an organic compound and it is tightly bound to the apoenzyme, organic compound and tightly bound to the apoenzyme, then it is called as prosthetic group. Main thing is how it is bound. It should be tightly bound. For example, heme is a part of the active site of enzyme catalase and peroxidase. Okay. So that heme is the prosthetic group because it can never be separated. If you separate, the enzyme will lose its activity. So organic compound firmly bound, tightly bound to the apoenzyme is called as prosthetic group. Second is coenzyme. Coenzymes are also organic compounds. Mostly they contain the vitamins, and these are transiently bound to the enzyme. Transiently bound means during enzyme catalysis they will bind and they will separate. You see, NAD. This is a coenzyme. Why? Because suppose pyruvic acid has to get converted into acetyl-CoA. NAD will come here, catalyze the enzyme, convert it into NADH, and this NADH will not remain bound to the enzyme. It will separate and it will go to the inner mitochondrial membrane to get reoxidized. So this NAD bound to the enzyme pyruvate dehydrogenase only during enzyme catalysis. Once catalysis was over, it separated from the enzyme, went to the inner mitochondrial membrane, get oxidized to produce ATP. So organic compound mostly containing vitamins and transiently bound to the apoenzyme only during enzyme catalysis, these are called as coenzymes. So main difference between prosthetic group and coenzyme is tightly bound prosthetic group, transiently bound coenzyme. Last is the metal ions. Okay. Third one is the metal ions. A number of enzymes require metal ions for their activity, which form coordination bonds with side chains of active site. Okay. For example, zinc is a cofactor for the proteinaceous enzyme carboxypeptidase. This metal ion, when it functions as the cofactor, it forms coordinate bond with both, with the apoenzyme also and with the substrate also. See here. which form coordinate bond with the side chain of the active site, means of the apoenzyme, and at the same time forms one or more coordinate bond with the substrate also. Zinc is a cofactor for the proteinaceous enzyme carboxypeptidase. A very other example, if you are asked, a very good example is molybdenum. Molybdenum is a cofactor of the enzyme nitrogenase. This is also. Any trick for the types of enzymes? एक word है. Someone is asking any trick for the types of enzyme. There is a word, if you can remember. Oxalil. If you can remember this word, oxidoreductase first class, transferase second class, hydrolase third class, lyase fourth class, isomerase fifth class, and ligase sixth class. So if you can remember this word oxalil, it can be a bit easy for you. Otherwise, I do not much believe in this shortcuts. Okay, fine. So children, with this we have completed this portion. That is the biomolecules. Next we are coming to cell cycle and cell division. Let us see. Just a water break. Cell cycle and cell division. Fine. Okay. If you see this diagram, see. Remember, Rudolf Virchow said that the new cells arise from pre-existing cell. So how from pre-existing new cells arise? This is a cycle of that. Okay. See here. So a cell division cycle mainly consists of two phases. This particular portion, this is called as the interphase. This complete blue line that you see, this is called as the interphase. And where actually cell is dividing, this is called as the M phase. Interphase is the duration where the cell is getting ready to get divided. Okay, the cell is getting ready to divide it. It has been divided into three parts, G1, S, and G2. During G1, most of the cell organelles are divided, multiplied, okay, duplicated. During S phase, there is synthesis of DNA. As a result, the DNA content of the cell becomes double. Okay, as a result, the DNA content of the cell becomes double. So initially there were amount of DNA, if initially it was 2C, by the end of the S phase it will become 4C. But remember, chromosome number does not increase. So if the start of S phase it was 2N, at the end it will also remain 2N. Okay. So chromosome number does not increase, but DNA content becomes double. For this NCERT in the summary has written chromosomal duplication. So we think that chromosome number is duplicating. No. Chromosomal duplication means that at the start of S phase, each chromosome had only one DNA. And at the end of S phase, one more DNA is added to each chromosome. So you see, chromosome number did not increase, but DNA content become double. That is why the 2C remains, the 2N remains 2N, but the DNA content from 2T to 4C, because one more DNA is added to each chromosome. The chromosome number remains same, but the DNA content each chromosome becomes double. I think this point is clear to everyone. Hello. And apart from that, during S phase also, in the cytoplasm of animal cell, there is duplication of the, in cytoplasm, thank you. In the cytoplasm of the cell, there is also duplication of the centrosome. So this additional things takes place during S phase. Now G2 phase, further growth of cell continues and RNA and protein synthesis continues, but there is no DNA replication. Now the cell is ready to divide. The division phase is called as M phase, and this M phase is completed in 4: prophase, metaphase, anaphase, and telophase. So this 4 is related to nuclear division. Okay, this 4 is related to nuclear division, which is called as karyokinesis. And this is the division of cytoplasm, which is called as cytokinesis. One more thing, children, if the cell decides I will not divide further, for I will go to a permanent rest or temporary rest, then by the end of G1 phase, it should somewhere here. By the end of G1 phase, it comes out of the cell cycle and undergoes a resting phase, okay, a resting phase. This is called as the quiet phase or quiescent phase or G0 phase. Remember, in G0 phase, the cell will not divide, but it is metabolically active. It does not mean it has become dead. Metabolically active, but not divide. Now here the cell has to take decision. Either it may never come back to the cell cycle. For example, the heart cells. Or if required, they can come back. For example, when there is dedifferentiation of the permanent tissue and again they become meristematic. So it is the decision of, that is why we say a cell in G0 phase is in suspended phase. It may or it may not, it may come back to cell cycle, it may not come back to cell cycle. This depends. Is it clear? Fine. Now children, the interphase we have completed. Now we come to the M phase and we are going to study mitosis. Mitosis is a type of cell division. Mitosis is a type of cell division where a mother cell divides to form 2 genetically identical daughter cells. Okay, both daughter cells have same number of chromosomes, same genetic constitution. Questions in the corner of the page below the diagram. Which are the questions? Acha, how do plants and animals continue to grow all their lives? By mitotic cell division. Okay, plants have meristematic tissue. Plants have meristematic tissue. Okay, these meristematic tissue are active throughout the life. They keep on dividing. Animals do not grow throughout their life. After some time, their growth stops. But some dead and worn out cells are replaced, and again, this is done by mitosis. Fine. Do you think all cells continue to divide, all plants and animals? No. The cells that convert into permanent tissue, they may not divide. Similarly, in animals also, after some time cell division, once the growth is completed, cell division stops. Okay. Tissue having cell that divide all their life in the hand. Yeah. In higher plant, there are meristematic tissue at the root and shoot apex. They divide throughout their life. Do animals have similar? No. Animals don't have meristems. So animal, that is why animal growth is called as determinate growth. But yes, animals have such provision that dead and worn out cells, for example, the intestinal lining gut cell, the epidermal cells, the RBCs, they are continuously replaced. Although growth as such is not taking place, but they have ability to replace these cells. Okay, fine. This question is done, which is the other question. Acha. You have studied mitosis in onion and root tip. It has 16 chromosomes in each cell. Can you tell how many chromosomes will be there in cell in having G1? Chromosome number in mitosis does not change. So whether G1 or G2 or whatsoever, it will remain 16 only. Okay. DNA content in G1, if it is 2C, S phase and most of the M phase, it will be 4C. Okay. S phase and most of the M phase, it will be 4C. Okay. Can you tell how many chromosomes will be there in cell have G1 phase after? And also what will be the DNA content of the cell at G1? G1, if it is 2C, after S, it will 4C, after G, it will also be 4C. If the content after M phase is 2C, No, the content after M phase, yes. After cell division, the content will again reduce to 2C. Let us first complete the mitosis, then you will understand. Fine. Okay. Any other question? I think that's all. Okay, ji. Haan ji. As I said, the M phase, since it is actually division phase, that is why it is called as the most dramatic phase of cell cycle, and it is divided into 4 subphases: prophase, metaphase, anaphase, and telophase. During prophase, children see here, in interphase, the chromatin is not condensed to form chromosome. So prophase, first of all, the chromatin condenses to form chromosome. So individual chromosomes become distinct. Each chromosome has 2 sister chromatids, fine, connected through a centromere. And as the prophase is ending, the nuclear envelope, the nucleolus, the mitochondria, and the chloroplast, they start disintegrating. This is the end of the prophase. So in prophase, chromatin condenses to become distinct chromosomes, and by the end of prophase, nuclear membrane, nucleolus, endochloroplast, endoplasmic reticulum, they all, okay, endoplasmic reticulum, nucleus, and the nuclear envelope, Golgi complex, they all disintegrate. Now, next phase is called as the metaphase, but there is a phase between the prophase and metaphase which is called as transition to metaphase. Okay, then we come to metaphase. In metaphase, you can see the centrosomes or the centrioles, they have formed the spindle apparatus. Okay, we have already said that the centrioles are important for the formation of a spindle apparatus during the cell division. The spindle apparatus contains an aster. This structure is called aster, and from the aster, the radiation of the microtubules. Each one is attached to the kinetochore of the chromosome. So each chromosome is attached to 2 spindle fibers coming from opposite direction. So first of all, formation of the spindle apparatus is completed. Okay. Okay. The formation of the spindle apparatus is completed. Next, the chromosomes show maximum condensation. Video stuck. Acha, रुको. Please call the technical person. A video problem. They are saying video is stuck. ये ऑन कर दो. Okay. So, okay, fine. So metaphase, 3-4 things we have to remember. First of all, spindle apparatus formation is completed and the kinetochores of the chromosomes get attached to the spindle fibers. Each chromosome is attached to 2 spindle fibers coming from opposite sides. Second, chromosomes are completely condensed. That is why best visibility. That is why this is the best phase to study the chromosome morphology, like the chromosome morphology numbers. This is the best phase to study. This is the best phase to study metaphase. Okay. Now, and third thing is all the chromosomes are attached to an equatorial plate, which is called as the metaphasic plate, and this completes the metaphase. Fine. Okay. Next is, children, very important thing is now the chromosomes will, the sister chromatid will separate due to splitting of centromere. This question is very frequently asked, 2 years back also asked. In which phase there is splitting of the centromere? So remember, anaphase of mitosis and anaphase second of meiosis. Fine. So next phase is anaphase. You can see here the centromere splits, so the sister chromatids are separated and they move to opposite poles. This is called as anaphasic movement. These two, which were till now sister chromatids, these were till now sister chromatids of one chromosome. Now they are called as daughter chromosomes. So they are now called as daughter chromosome. That is why now you see the number of chromosome in the cell for a short period became double. That is why sometime anaphase is called as the polyploid phase of cell cycle. You see here, there were 4 chromosomes, 1, 2, 3, 4. Now at each pole there are 4 chromosomes, means total 8 chromosomes. That is why for a short period it becomes a polyploid phase of the cell cycle. Now anaphase, by the end of anaphase, at each pole of the cell there will be group of chromosomes, and number of chromosomes in each group will be same as present in the mother cell. Now last phase comes, that is the telophase. During telophase is almost opposite of the prophase. First thing is chromosomes will decondense and lose their identity. Second, there will be formation of the nuclear envelope around both the groups of chromosomes. As a result, nucleolus and the Golgi complex and endoplasmic reticulum will reappear. Okay. So within a single cell, 2 nuclei are formed. This completes the karyokinesis. Okay, division of the nucleus. But actual division will take place when cytoplasm will also divide. This is called as cytokinesis. Okay. Cytokinesis, children, is of 2 types. In animal cell, where there is no cell wall, simply you see here, you can see what happens. This is a constriction, a furrow is formed. So in the cytokinesis of animal cell is called as the cleavage furrow formation method. Okay, cleavage furrow formation method, where there is a constriction in the cytoplasm and ultimately the mother cell divides to form the 2 daughter cells. But remember, in plant cell, there is a rigid cell wall. So plant cell cannot undergo cleavage furrow formation. So in plant cell, what happens? Suppose these 2 nuclei are formed. In the center, a cell plate is formed. For this formation, Golgi and vesicles are important. Vesicles of Golgi body, they come here, assemble, and then there is deposition of the calcium pectate. This cell plate is formed and it grows centrifugally. This grows cell plate ultimately grows and it divides the cell into daughter cell. So in plant cell, cytokinesis is by cell plate formation, and in animal cell it is by cleavage furrow formation. As a result, a mother cell divides into 2 daughter cells. Now what is the significance of mitosis? Let us see here. The significance of mitosis is Mitosis usually results in the production of diploid daughter cells with identical genetic constitution. So it means it maintains same genetic constitution. If the content of DNA in a chromosome is 2C, after M phase, what will be the content of DNA? You are asking about a cell or a chromosome? In a DNA at G1, G1 2C, at the end of S 4C, anaphase of mitosis, it will still remain 4C, and when there will be cytokinesis, then it will again revert to 2C. That is why this question is asked. So 4C starts from the end of S phase and it continues up till the cytokinesis. Up till the karyokinesis. Once cytokinesis takes place, it again converts into 2C. Okay, fine. Second point, growth of multicellular organism is due to mitosis. So growth. Third, cell growth results in disturbing the ratio between nucleus and cytoplasm. It therefore becomes essential for cell to divide to restore. So third is to restore the ratio between the nucleoplasm and cytoplasm, which is called as nucleocytoplasmic index. A very significant contribution, fourth point C, of mitosis is cell repair. Because as I said, many cells, for example, gut lining cells, you know, in a stomach, there is production of HCl and it burns the cells of the gut lining. So it has to be very rapidly replaced. Skin cells, very rapidly they are replaced. Okay, there. So that mitosis helps in replacement of the dead cell. This is called a cell repair. Suppose we get a wound, a cut, ultimately it is healed up. How? It means mitosis taking place here for repairing. Okay, so cell repair. Okay. Cells of the epidermis, gut lining, blood being constantly replaced. Mitotic division in the meristematic tissue. See, due to mitosis in the meristem, the apical meristem, the plants keep on growing throughout their life. So this is also due to mitosis. So these are the children advantages of mitosis with which finish mitosis. Next, we are coming to meiosis. Interphase is similar. There is no difference in the interphase of mitosis in meiosis. Okay, but meiosis has two cell divisions. First one is called as the reductional division and second one is called as equational division. Meiosis always takes place in diploid cell only. Mitosis can take place in diploid and haploid. In animals, generally in diploid cell. In plant, both diploid and haploid. But meiosis takes place only in diploid cell and it takes place only in sexually reproducing organism. There is no sexual reproduction, there will be no meiosis. Okay, fine. Now, I am giving you a chart. Please see, this is very, very important to understand the things. Meiocyte after S phase. Meiocyte means the cell which has to undergo meiosis. After S phase means DNA content has become double. DNA content has become 4C. Okay. Suppose 2n is equal to 20 and DNA content is 100 picogram. Suppose DNA content is 100 picogram. Fine. Now, meiosis first or reductional division. Each daughter cell will receive half. So n is equal to 10 and DNA content is equal to 50 picogram. N is equal to 10 and DNA content is equal to 50 picogram. Fine. Now both daughter cells will divide and this is just like mitosis. You remember in mitosis, DNA content becomes half, but the chromosome number does not change. So here n is equal to 10 and DNA content will be reduced to 25. N is equal to 10, DNA content will be reduced to 25. N is equal to 10, DNA content will be reduced to 25. N is equal to 10, DNA content will be reduced to 25.
Okay, this is called as meiosis second or equational division, because chromosome number does not change, does not reduce. That is why it is called as the equational division or meiosis second. So meiosis first, since chromosome number is reduced to half, it is called as reductional division, and meiosis second, chromosome number remains same, that is why it is called as equational division. But from here to here again, DNA content will be reduced to half. So if a gamete has 10 picogram DNA, it means the meiocyte would have 40 picograms DNA. Okay, remember this point. So if a gamete has 10 picogram DNA, means the meiocyte would have 44 times. Because you see, this 100 is the 4 times of this 25. Okay, now coming to meiosis first. Meiosis first is a very long process. That is why it has been divided into 4 subphases. Please, I am telling you, remember these points. First, meiosis first one is called as the leptotene. During leptotene, the chromatin material condenses, so individual chromosomes become distinct. Each chromosome has 2 sister chromatids, and the cell has 2 sets of chromosomes. Okay, one maternal set of chromosome, one paternal set of chromosome. One set is also called one genome. For every chromosome, there is an identical another chromosome. For every chromosome of maternal set, there is an identical chromosome of paternal set. These 2 chromosomes are called as homologous chromosomes. So there are, okay, homologous chromosomes, 2 sets of chromosomes. Okay, leptotene, after leptotene, there is zygotene. During zygotene, there is formation of a structure called as synaptonemal complex. This synaptonemal complex causes pairing of the homologous chromosomes. This pairing of homologous chromosomes is called as synapsis. Okay, so this is important, that zygotene, there is formation of the synaptonemal complex, and synaptonemal complex causes pairing of homologous chromosomes, and this pairing of homologous chromosomes is called as the synapsis. Fine. Next. Now, next comes is the packetine. This paired homologous chromosomes are called as bivalents. They are also called as tetrad. But at this time, tetrad condition is not very clearly visible. Next comes the packetine. During packetine, first of all, the tetrad condition becomes visible. We can see they are, each bivalent consists of 4 chromatids. Next thing is that on the bivalent, there will be formation of an enzyme complex which is called as recombination nodule. This recombination nodule contains an enzyme recombinase. With the help of this enzyme recombinase, there is exchange of chromatid segments between homologous chromosomes, and this phenomena is called as crossing over. So very important feature of the packetine is crossing over. Due to crossing over, new combination of genes is formed, which is called as a genetic recombination, Fine. Okay. Now diplotin. Once the crossing over is over, the pairing between homologous chromosome ends, and the homologous chromosomes start to separate from each other. Okay, and they start to separate from each other. Okay, this question is also very frequently asked: at which phase separation between homologous chromosomes starts? Chiasmata is Arey bhai, thoda Jagan, please have some patience. Okay, so when the homologous chromosomes separate from each other in the diplotin, you will see at certain places where crossing over had taken place, at those places they still remain attached to each other. There you will see a cross-like structure which is called as chiasmata. So remember, crossing over in packetine, okay, but the chiasmata formation you will see in the diplotin. Okay, now further repulsion between homologous chromosomes will take place, and this will come to the last phase of the prophase first, which is called as the diakinesis. During diakinesis, the homologous chromosomes are attached only through the terminal chiasmata. This movement of the chiasmata from the intercalary position to the terminal position is called as terminalization. So important feature of the diakinesis is terminalization. Fine. Now we are coming to the end of prophase 1. So there will be disappearance of the nuclear envelope. Okay. Nuclear envelope, as you saw in case of mitosis, ER and all that. Fine. Okay. So now the situation is at the end of prophase 1, the homologous chromosomes are attached only through the terminal chiasmata. Fine. Just wait. Now see this diagram carefully. During the interphase, there was crossing over. As a result, you see there is exchange of chromatid segment between homologous chromosomes, which you can see here. Now, at metaphase, what will happen, children? At metaphase, what will happen? These paired homologous chromosomes, they attach at the spindle. So metaphase 1, the bivalents attach at the metaphasic plate, okay? And each chromosome is attached only to one spindle fiber. Now anaphase 1. This diagram is of anaphase 1. The terminal chiasmata now also breaks and the homologous chromosomes are separated and moved to opposite poles, okay? This is anaphase 1. Remember, in anaphase 1, there is no splitting of the centromere. In anaphase 1, there is only separation of the terminal chiasmata. So the terminal chiasmata breaks and the homologous chromosomes separate to opposite poles. Now you see, you started with 4 chromosomes, but each pole has only 2 chromosomes. It means chromosome number will, in the daughter cell, be reduced to half, okay? Chromosome number in the daughter cells will be reduced to half. See this NCERT diagram. See, prophase 1 crossing over completed. Metaphase 1, homologous chromosomes attached to the metaphasic plate. But you can see here, due to crossing over, you see this color change you can see due to crossing over. Fine? Okay. Anaphase, homologous chromosomes separate and move to opposite poles. And once homologous chromosomes before crossing over are, they may not be genetically identical. Maybe on one you have capital T, on other you have a small t. That may be heterozygous condition, it may be homozygous condition, you cannot say. Jagan, what is wrong? Okay, fine. So after meiosis, first there will be 2 daughter cells. After meiosis, there will be 2 daughter cells. Each daughter cell will contain half the number of chromosomes. Remember, you started with 4 chromosomes, but each daughter cell has only 2 chromosomes. Fine. So that is why we call it as reductional division. And you see, now the sister chromatids are not genetically identical because in one of them crossing over has taken place. So this is meiosis first. After meiosis first, both the daughter cells will undergo mitosis. Meiosis second, which is exactly like mitosis. This is called as a dyad of cells. This is 2 cells, which is called as dyad of cells. Now, both daughter cells are undergoing meiosis second. Meiosis second is just like mitosis. As a result, we will get a tetrad of cells. Each cell will have 2 chromosomes, while we started mother cell with 4. Okay, so this is called as the meiosis second. Okay, now what is the significance of meiosis? Let us try to see. First of all, crossing over has taken place. Due to crossing over, new combination of genes are formed, means genetic variation will take place, and this genetic variation is very important for evolution. So first of all, crossing over, genetic variation is responsible for evolution. Second is Jagan, don't read such books which write chiasmata is formed in packetine. Read your NCERT and start reading some books. Better confine yourself to your NCERT, otherwise you will suffer. Okay. Sometimes children over reading also becomes a problem. We have a very basic syllabus, but if you start reading international journals, then it is a problem. Don't do that. Okay, yes. Second is, children, although meiosis is reducing the chromosome number to half, but it helps in maintaining constant chromosome number in a species generation after generation. How? Let us try to understand this. Suppose human male
You know 2n is equal to 46. So if gamete is formed, sperm is formed without meiosis, then sperm will have 46 chromosomes. And similarly, human female, 2n is equal to 46. If egg is formed without meiosis, then egg will also have 46 chromosomes. And if when they will undergo fertilization, then the zygote will have 4n is equal to, I think, 92. So does this happen that every next generation chromosome number becomes double? Okay. Does this happen that every next generation chromosome number becomes double, or chromosome number remains constant generation after generation? Anaphase of mitosis, anaphase 2 of meiosis have no difference. But the only difference is that in mitotic anaphase, the two sister chromatids that are separating, they are genetically identical. In anaphase second of meiosis, the two sister chromatids that are separating, they are genetically non-identical due to crossing over earlier. Okay. Clear. Fine, children. But this does not happen, remains constant. Why? Because this male forms gametes by meiosis. So gamete is, the sperm is 23. Similarly, meiosis. So egg has 23. Now when there will be fertilization, the zygote will have again 2n is equal to 46. So although meiosis reduces the chromosome number to half, but it helps in maintaining constant chromosome number in a species generation after generation. Let us see this point. See, paradox means self-contradictory. See, even though the process per se paradoxically results in reduction of chromosome number by half, it also increases the genetic variability. Okay, fine. Conservation of a specific chromosome number of each species is achieved across generation in sexually reproducing organism, even though the process per se paradoxically results in reduction in chromosome number by half. So they are saying that it is just a self-contradictory, but it is true that meiosis itself reduces the chromosome number to half, but it helps in maintaining constant chromosome number in a species generation after generation. Because if this does not happen, then in every next generation chromosome number will become double. So children, with this we have completed the cell division and cell cycle. So this portion is completed. Okay. So now we are left with just half an hour of time, and we have physiology. So let us start with photosynthesis, and then we will see how much we can carry on. Fine. Okay. Okay. So synthesis of organic food. By green parts of the plant by combining CO2 and water in presence of sunlight. This is called as photosynthesis. Fine. Okay. So it is an phototrophic. Now, if you see photosynthesis as a phenomena, it is an anabolic process because there is synthesis of food. It is an endergonic process because there is energy is being used, sunlight energy. So anabolic process, endergonic process. Next, it is oxidation reduction process because water is oxidized and CO2 is reduced. So it is an oxidation reduction process, and it is directly or indirectly source of food for all living organisms on earth. And it also provides oxygen because the oxygen that is released that helps the organisms to oxidize the food. So this is all regarding the mechanism. Fine. Now, historical aspect, I'm not too much going. You have studied that Jane Ingenhouse, Joseph Presley said that plants purify air. Jane Ingenhouse said that green parts of the plant only can purify air, and the gas released is the oxygen gas. Then Julius von Schacht said that the green pigment is important for photosynthesis. Plants synthesize food as glucose and store as a starch. Then T.W. Engelmann gave the action spectrum of photosynthesis, where he said that photosynthesis, maximum photosynthesis takes place in blue and red light. Okay. And then it was proved by Van Niel that the oxygen released in photosynthesis comes from water and not from the CO2. These are the basic aspects. Okay. Now coming to the photosynthetic apparatus. If you see the photosynthetic apparatus, this is the chloroplast, which is the photosynthetic apparatus. It is a double membrane bound structure. Interior, the diagram I've already told you. Here main thing is the interior of the chloroplast has two parts. The fluid part is called as the stroma, which is the site for dark reaction of photosynthesis. Okay. And these grana and stroma lamella together, these are the site of light reaction of photosynthesis. Okay. The product of light reaction of photosynthesis are basically ATP and NADPH. These are the site of light reaction of photosynthesis. These are the products of light reaction of photosynthesis. Fine. Now, these grana and stroma lamella are together made up of thylakoids. In these thylakoids, there are photosynthetic pigments to trap the sunlight. These photosynthetic pigments in higher plants are of following types. Chlorophyll A, chlorophyll B, xanthophylls and carotenes. Okay, so these are the four types of photosynthetic pigments. Chlorophyll A is the chief photosynthetic pigment, main photosynthetic pigment. All others are called as accessory pigments. Chlorophyll A is bluish green in color. Okay. Chlorophyll A is bluish green in color. Chlorophyll B is yellowish green in color. Carotenoids are orange to yellow orange, and xanthophylls are yellow in color. Okay, these different pigments, they absorb different wavelength of light. So this diagram which is shown here, this is called as the absorption spectrum. This is called as absorption spectrum. This diagram shows which photosynthetic pigment absorbs which light maximum. So if you see, the green is of chlorophyll B. So chlorophyll B absorbs maximum blue light, and again
red light. So chlorophyll B absorbs blue and red light maximum. Similarly, if you see this blue is of chlorophyll A, same chlorophyll A also absorbs maximum blue light and then red light. But as compared to chlorophyll A, chlorophyll B absorbs more blue light. But as compared to chlorophyll B, chlorophyll A absorbs more red light. Okay, so this is called as absorption spectrum. And the wavelength of light which is not absorbed by these two are absorbed by carotenoids. So carotenoids function as accessory pigment. They use that wavelength of light also in photosynthesis, which are not used by chlorophyll A and chlorophyll B. Okay, fine. And one more thing, carotenoids, they protect the chlorophyll from photooxidation, means in very high light intensity, chlorophyll gets oxidized and damaged. So carotenoids have two functions. First, to function as accessory pigment and use that light in photosynthesis, which chlorophyll cannot use. And second, to protect the chlorophyll from photooxidation. See here. See, they not only enable a wide range of wavelength incoming light to be utilized for photosynthesis, but also protect chlorophyll A from the photooxidation. Now, second diagram, if we see here, children. This is called as the action spectrum. Action spectrum means that in which light, how much photosynthesis takes place. In which light, how much photosynthesis takes place. Now, if you see here, children, that maximum photosynthesis takes place in blue and red light. Blue and red light. This was discovered by T.W. Engelmann. Now we are comparing the absorption spectrum of chlorophyll A with the action spectrum of photosynthesis. So you will see a very striking similarity. The light that is absorbed maximum by the chlorophyll A, that is blue and red light, in the same light maximum photosynthesis takes place. Okay, so this way we can say that the main or chief pigment involved in photosynthesis is chlorophyll A, because the light which chlorophyll A absorbs maximum, in the same light maximum photosynthesis is taking place. Okay, so this is proof that the chief photosynthetic pigment is chlorophyll. See here. Can you see that the wavelength at which there is a maximum absorption by chlorophyll A, that is in the blue and red region, are also show high rate of photosynthesis. Hence, we can conclude that the chlorophyll A is the chief pigment associated with photosynthesis. Okay. Now, children, these pigments are not randomly present. These pigments are present these pigments, suppose this is a thylakoid membrane. These pigments are present in the form of protein pigment complexes. These protein pigment complexes are called as photosystems, PS, photosystems or PS. So in the thylakoid membrane, the pigments are present in the form of protein pigment complexes called as pigment system. Now, if you see each pigment system, each pigment system consists of around 250 to 400 pigment molecules. Now, each pigment system has two components. If you see this one, this single chlorophyll A molecule is called as reaction center. All other molecules are called as antenna molecules. Okay, and they just help the chlorophyll A. So a pigment system has two parts. A reaction center, which is a chlorophyll A molecule, which is absorbing light of maximum wavelength. Another one is called as the light harvesting complex or antenna molecules, which contain all other pigments. When light falls, the antenna molecules also absorb light and get energized. Reaction center also energizes. Now the antenna molecules, they transfer their energy to the reaction center. Okay, you can see this. So antenna molecules transfer this energy to the reaction center. And reaction center gets so much light energy that it loses an electron to a primary acceptor. And this electron now moves in an electron transport chain, which is called as the ETS. Now, it was discovered that there are two types of pigment systems in the plant. Okay, there are two types of pigment system called as PS1 and PS2. So one that was discovered earlier was named as PS1 and one that was discovered later was named as PS2. So earlier and discovered later. Its reaction center is called P700 because it can absorb light of up to 700 nanometer. This reaction center is called P680 because it absorbs light of 680 nm. So beyond 680 nm, PS2 cannot function because the reaction center is P680. Now, this PS1 is involved in two types of transports. PS1 is involved in non-cyclic electron transport system as well as cyclic electron transport system. And PS2 involved only in non-cyclic. Now PS1, where it is present? PS1 is present in stroma lamella and appressed part of grana. Sorry, non-appressed part of grana. Stroma lamella and non-appressed part and this is present, PS2 is present in appressed part of grana. Now you will see what do you mean by appressed and non-appressed part. So I will show you, see here. Here PS1 will be found, only PS1. PS2 is absent in stroma lamella. Here also PS1 will be found. Means the free surface. Free surface is called as non-appressed part. PS1. And this attached surface here PS2 is present. Is it clear? Who was asking this question? Sai Adarsh, is it clear now? Okay, fine. Now, let's see what is light reaction. Let us try to see this. See here, there are, as I said, there are two types of pigment system, PS2 and PS1. When both are involved, the electrons do not return back to their original donor. That is why it is called as non-cyclic electron transport system. So what happens? That light falls on both PS2 and PS1 simultaneously. In PS2, the reaction center loses an electron to a primary acceptor. Okay, primary acceptor. This is called as uphill transport. Now downhill transport starts. Okay, that downhill transport starts, and for this electron carriers are arranged in the increasing order of the redox potential from lower to higher redox potential. This natural movement is called as the downhill movement. Okay, because energy wise it is coming downhill from higher energy to lower energy. That is why it is called as downhill movement. But if you say redox, then it is from lower to higher redox. But this is called as downhill movement. Fine. Okay. Now the primary acceptor here is phaeophytin, which gives electron to acceptors. Similarly, the PS1 has also lost electron to a primary acceptor. Okay. And this electron is taken by NADP plus, which gets reduced to NADPH. So one of the products of light reaction, NADPH, are formed. Now the PS2, the electron that PS2 has lost to PS1, this electron it fulfills from water. So that is why at PS2 there is a splitting of water. Okay, there's splitting of water to fulfill this electron, and this oxygen is released as one of the final products of the light reaction, which we see in the atmosphere. Fine. Not only this, children, during this downhill journey of electron, there is sufficient energy available for production of ATP. So remember, we said there are three products of light reaction: NADPH, ATP, and oxygen. All are produced here. This is called as non-cyclic photophosphorylation because the electrons released from the PS2 never return back to the PS2. That is why it is called as non-cyclic. There is one more method also which is called as cyclic photophosphorylation. Cyclic photophosphorylation involves only PS1. There is no PS2 involved. There is no water splitting. There is no NADPH formation. There is only ATP synthesis. Why it is called cyclic? Because you see electrons coming out of PS1 finally return back to PS1. Okay, that is why there is cycling of electron. That is why called as cyclic. There is no need for water splitting. No NADPH formed. Okay, but children, this electron transport, how it is involved in ATP synthesis, the actual mechanism is called as chemiosmosis. So let us try to see this chemiosmosis. Because this electron movement directly does not produce ATP. Electron movement creates a proton gradient across the thylakoid membrane. And when this proton gradient is broken down, then there is synthesis of ATP. How proton gradient is created across the thylakoid membrane? Let us try to see. First of all, when the PS2 loses its electron to an acceptor here, plastoquinone, this plastoquinone also accepts a proton. And this proton, which it is accepted from stroma, it pumps it into the thylakoid lumen. So this is called pumping because it is energy-oriented process. As a result, in the stroma, there is shortage of protons, and in the thylakoid lumen, there is increase of protons. Second thing is that when there is shortage of electron to the PS2, it splits water. So electron is taken by PS2, but protons are left into the thylakoid lumen. So this is the second reason of the creation of proton gradient across the membrane. Third is, at PS1, when NADP is reduced to NADPH, the protons are taken from the stroma. So you see in a stroma, there is continuous shortage of protons because proton from here pumped here and protons used for reduction. While in lumen, there is increase of proton, first due to this pumping and second due to release from the. As a result, high electrochemical potential gradient is created across the thylakoid membrane. Now, and remember, this was created due to ETS. That is why ETS is important to create the proton gradient. Clear? Now, how this proton gradient helps in ATP synthesis? Remember, children, proton is charged particles. And remember, we have said in today's lecture only that lipid bilayer of membrane does not allow any charged particle to pass through it. It means charged particle, that is a proton, can only pass through the membrane protein. Here, there is a complex which is called as ATP synthase complex or CF0-CF1 complex. CF0 is an integral membrane protein embedded in the membrane, and CF1 is a peripheral protein attached to CF0. Now this CF1 has ATP synthase enzyme, but it is in inactive form. Now what happens? The CF0 is a tunnel protein that provides facilitated diffusion to protons. When protons undergo facilitated diffusion, there is a conformational change in the CF0. Okay. As a result, the CF1 becomes active. So movement of protons through the CF0 activates the CF1. And CF1 has ATP synthase, with the help of which it is able to produce ATP. It means the final cause for ATP synthesis is breakdown of the proton gradient across the membrane. But for breakdown of the proton gradient, first there should be a creation of the proton gradient. To create the proton gradient, there should be electron transport. And to initiate electron transport, there should be light absorption. So now let us see the sequence of events during light reaction. Absorption of light by pigment systems, excitation of the chlorophyll, loss of electron to a primary acceptor, electron moves in electron transport chain. Due to electron transport chain, there is creation of a proton gradient across the membrane. due to this breakdown of the proton gradient through CRF0, CRF1 complex, and due to breakdown of the proton gradient, there is synthesis of ATP. Okay. It means the important thing that are required, this method of ATP synthesis is called as chemiosmotic hypothesis. Okay, now let us see that it means for chemiosmosis, the things that are required are see, first of all, a membrane across which there will be proton gradient creation, a pumping of protons which will create a proton gradient, a third a proton gradient, and breakdown of proton gradient for this ATP synthesis. So there are 4 basic requirements for this. First of all, there should be a membrane. Second, there should be pumping of protons to create a proton gradient. Third, there should be a creation of proton gradient. And fourth, there should be an ATP synthesis. So when the proton gradient is broken down, there is synthesis of ATP. And along with that, there is also synthesis of NADH that you have seen here. Please see. Okay, fine. So ATP also synthesized, NADPH also synthesized, and the third product of the light reaction, that is oxygen released. So there are 3 products of light reaction: ATP, NADPH, and oxygen. Oxygen are released in the atmosphere, but ATP and NADPH are immediately used in the dark reaction for the synthesis of the sugar. But children, that is all we have time for today. Okay, if there is any second part, I will let you know. Otherwise, that's all. Thank you. Bye bye. God bless you.